My name is Clive Roper. I am one of the directors of the North American 3Rs Collaborative, and I'm here just to welcome you today. Megan will be joining us as well later on. So the North American 3Rs Collaborative and IQ Microphysiological Systems webinar and workshop series, and today we're going to be talking about liver models, and welcome from the North American 3Rs Collaborative. I've got to give a disclaimer here. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussions of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither North American 3Rs Collaborative nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during the collaboration activities. So for the introduction to today's meeting, this webinar, introduction to the North American 3Rs Collaborative IQ in this webinar workshop series. So the North American 3Rs Collaborative mission is to advance science, innovation, and research animal welfare. We are without doubt refine, reduce, and replace. Our Microphysiological Systems initiative seeks to increase adoption and regulatory acceptance of microphysiological systems, but in coordination with animal studies. These are not separate, there's still integrated toxicology to be considered here. We have 39 institutions, including 28 developers are the members. I'm not going to take you through all the names here, but you can see we've got a wide variety from multinationals through to very small spin-out companies, and we also have the FDA centered in the middle here as part of our groups. We are hosting a series of microphysiological system focused webinars and workshops. The webinars are company and system independent and focused on general education about an MPS system or topic. The workshops are company specific and focused on presenting information about commercially available MPS systems. You'll hear from 9 companies across the 2 days, today and tomorrow. We'll start off with CN Innovations, then Draper, Emulate, Hesperos, and InSphero, and then we'll have a short roundtable discussion. Tomorrow again, I'll be introducing, and we will then follow up with In Vitro ADMET Laboratories, Discovery Life Sciences, Lena BioSciences, MicroMatrices, and TissUse, followed by another roundtable discussion. We will be keeping very much to time, so if you hear me, speakers, that's because I'm warning you. So with the questions, each company gets 15 minutes in total, including a brief Q&A period. We will be strict on time. If we get to 15 minutes, I will be shouting. Hopefully we'll have a short 1 or 2 questions after each presenter. The final roundtable can be used to ask questions of multiple companies or specific companies. So please be direct in your question. I guess if you're not direct and you don't put to a company or a name, then we're going to assume that that's a general question for the panel. Please submit your questions using the chat. The questions will be answered at the end of the webinar as time permits. So the chat there, and type in your comment, and attendees will be muted and have cameras off. So the only person you should be able to see is the presenter. Now let's briefly hear from the IQ and then our companies. So there should be an IQ slide to follow.
My name is Banu, investigator in GSK, but here representing the IQ MPS affiliate. So just wanted to take a minute, since we're already on time, to introduce or give an overview of our goals. So there are 4 goals here. First one is to serve as a thought leader for both MPS developers and stakeholder organizations in industry implementations and qualification of MPS models. Provide a venue for appropriate cross-pharma collaboration and data sharing to facilitate industry implementation and qualification of MPS. Now, create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS as an industry, and develop external partnerships and collaborations to enhance the inclusion of industry priorities. And like Clive also did, I'm not going to go over the member companies, but there are quite a few pharma companies listed here. And I just wanted to give a brief introduction to the 2022 leadership team, with the chair being Ree Hodwick and vice chair Anna Kopek and past chair Jason Eckert. With that, if you have any questions or suggestions or concerns, please contact the IQMPS at this address. And I can share my email address if you have any questions for the Strategic Communications Working Group. Happy to take them as well. Thank you. Back to you, Clive.
All right, thank you, everybody. Hopefully you can see my slides this morning. So thank you for the opportunity to speak to you. My name is Tom Koszczewski. I'm the head of science and technology at CM Bio. So it's a real privilege to talk to you today and tell you about how we've been building MPS systems, and we've built a number of different organ models, single and multi-organ MPS systems over the years. And today I'm going to tell you about the human liver model, which we've been developing over the last decade, and we really think this is one of the most advanced preclinical models of the human liver that's now available. So as a scientist looking for tools to study the human liver, there's now a wide range available, including MPS, and there's a broad toolbox for people to access. And we think that really the use of all of these tools in combination is key to ultimately getting translation, and it really depends on the question that people want to ask to find the right tool for them. But we think that the development of MPS has really added to this toolbox and is key to allow us to understand both liver disease, drug metabolism, and liver toxicology. And I'm going to show you how the MPS that we've developed allows you to look at all of those areas today. So when we built our liver MPS, and there was a number of key features and functional markers that we looked at, and actually these were all highlighted by the IQMPS in their sort of key paper series a couple of years ago. And I won't go through all the details, but again, I'll show you today how all of these key features of the human liver we recapitulate in our liver MPS around the production of biomarkers, the expression of appropriate enzymes, the expression of the right transporters and damage markers, etc. So our MPS technology is named PhysioMimics, and our liver MPS looks something like this. So our liver MPS is made up of, and I will just get my pointer up. It's made up of a 12 well MPS chip or MPS plate. So there are 12 individual microlivers on here. And if we zoom into one of those wells, we have a 3-dimensional collagen-coated scaffold on which we seed liver cells or combinations of liver cells into to form 3-dimensional microtissues. The micropumps embedded within these MPS plates perfuse cell culture media around in a circuit through this scaffold at all times, basically flowing oxygen, nutrients, and providing biomechanical stimuli to those liver cells. So we seed liver cells into this 3-dimensional scaffold as single cells. They form up over a couple of days into 3-dimensional microtissues, and then they're maintained highly functional and highly active in this 3-dimensional scaffold over a number of weeks. And so we form these column-like structures within these scaffolds. So we have hundreds of microtissues within each one of these scaffolds, and each one represents in some way the microarchitecture of the human liver that we're trying to replicate. And we have all the functional markers expressed of the human liver. So, for example, if we stain these microtissues for the production of albumin, you can see they stain it really beautifully here. We can see biocanaliculi networks form within these 3-dimensional microtissues. The cells form tight junctions. And so these liver MPS plates interface with our PhysioMimics technology, which I won't go through all the details of today for time. I'm going to focus on data. but basically we can one of our MPS systems can run multiple of these liver MPS plates, and this same liver MPS can be used as a single organ or as a multi-organ setup linked to other organs. And again, for today, I'm just really going to focus on how we use this system in a single organ context. So we typically run this liver MPS using primary human cells, but that's not the only option available. Other sources of cells are also usable, so we can put hepatic cell lines into this setup, and they form or stem cell derived materials, and they all form the same three-dimensional microtissues. We can use other animal species as well to form these tissues if we want to. And typically, the experiments we run are over a number of weeks. We think very hard when we run these studies, as I'll show you in a minute, that when we expose a therapeutic to these microtissues, that we do so in an in vivo, translationally relevant manner. And we can potentially induce a disease state within these liver microtissues if we want to. And a whole variety of endpoints can then be used at the end or throughout a study to understand the liver biology that might be changing throughout it. And a typical experiment in this kind of setup gives you an N number of, somewhere between 50 and 100 liver microtissues in a fairly typical study. And these are all very, very data rich. So we get multiple endpoints from every liver scaffold that we generate. We can typically run 2 or 3 cellular endpoints. So, for example, we can look at histology and the transcriptomics of the same microtissues from the same experimental setup. So we get incredibly data-rich experiments. So in this perfused MPS culture of primary liver cells, we have a really sort of functional system. We have a transcriptomic profile that, if we plug into an online database and ask it what it's looking at, it thinks it's looking at bulk human liver tissue, which is exactly what we want. We look at the abundantly expressed markers. We have all the key markers of hepatocytes being produced, those CYP enzymes, those transporters, etc. The biomarkers, we produce all the key biomarkers of the human liver, albumin, urea, etc. And we maintain the expression of key CYP enzymes, and these can be maintained across time. I'll show you a bit more of that in a minute. And this really is the basis for all of the different applications and context of use for how we can use this MPS system very straightforwardly. Just to dive into the biomarkers a little bit more, we actually produce the amount of albumin and the amount of urea that's principally produced by human liver. So typically we're getting greater than 100 microgram per million cells per day of urea, and greater than 25, 30 micrograms per million cells per day of albumin, which is again fairly equivalent to what is produced on a per cell basis by the human liver, and is the same as what was reflected in those criteria that I referred to on my early slides. So these three-dimensional liver microtissues can be formed of hepatocytes, but they can also incorporate all the other non-parenchymal and peripheral immune cells of the liver. So we can incorporate here Kupffer cells and stellate cells, and we can see the markers of those being detected in these microtissues. And we can add in sinusoidal endothelial cells or other peripheral immune cells if we want to, depending on the context of use and the scientific question that we want to ask. So we have a very modular setup, and we can add in different cell types if we need to, depending on the scientific question that we're looking to ask. So there's 3 main and key areas of focus for us when we use this liver MPS, and our partners use that, and that's in drug metabolism and disease modeling and liver toxicology. And I'm going to show you a little bit about each of these today, and how we really, the keys to use these successfully in these specific context of use. So to start off, I'll start with drug metabolism. So we take these three-dimensional liver microtissues, we'll expose them to a compound of interest to really understand the pharmacology of that compound, how it's broken down, what metabolites are being produced, and this is used, in a wide variety of different DMPK teams now. So we can use this, we can take the data that we generate from this liver MPS plate to understand the breakdown of the molecule, the production of certain, particularly human-specific metabolites, and we typically combine that with PBPK modeling to allow us to take the in vitro data of how a drug might be cleared and extrapolate that to get us in vivo relevant translational data. So to show you a little bit more about what that looks like, the liver microtissues, as I showed you, have all the a full variety of CYPs being expressed, so we can predict clearance rates really successfully if we compare the liver MPS here to more static conditions. This was data published by an external party, we see significant increases compared to more standard 2D hepatic cultures. And what that means is we can then get a really nice in vitro to in vivo correlation. Typically, the MPS just slightly underpredicts what is observed in vivo, but it's very, very correlative. And so we can do that extrapolation very consistently. This is data that we published a number of years ago. Further work has been done and actually was recently published by a pharma company, again, showing a wide variety of compounds and a really nice correlation. We can use the same setup to look at MetID, and so we can look at the breakdown of compounds. We can dig out the different metabolites being formed, and we can do this very simply. We can take multiple samples easily from our chips and our platforms to take temporal profiles to understand how these different metabolites get produced across time, and how that might relate to efficacious or toxicological effects. We have a wide variety of peer-reviewed publications in this area. I encourage you to kind of go and look those up through our website, etc. Show you a little about drug-induced liver injury now. So again, we can take these liver microtissues, we can expose them to a compound of interest, either for a single acute dose or very easily for a long-term chronic dose over a number of days. And we can look at a wide variety of endpoints to really interrogate what might be changing in the liver biology to understand the mechanisms of action of a particular compound. So first of all, I'll give you a classical example of how we might use this, being triglycerone. This is a molecule well known to fail in a wide variety of animal studies that failed to really predict its toxicity, and it was obviously withdrawn a number of years ago from the market. And if we put that same molecule into our liver MPS, we can generate standard IC50 type curves because we have a throughput to do that, and we typically will look at a wide variety of endpoints to really understand the mechanism of how this is causing toxicity. And importantly, we look at clinical biomarkers, so albumin and ALT, and for example, AST, to understand really how this molecule is causing toxicity to the human liver. We've now looked at more than 30 compounds to validate this assay. Many of those, nearly all of the compounds that were described in the IQ-MPS publication a number of years ago, we have tested and shown in the system. And we can very nicely correlate between compounds of high DILI concern and those with much lower DILI concern, and we get a great correlation across multiple endpoints. And I should say that all of these endpoints all get generated from the same well, same tissue. So again, we have a very data-rich setup. Finally, we can look at a variety of other endpoints related to DILI that are a bit more nuanced. We can look at transcriptomic endpoints. We look at how bile acid synthesis changes. So here's some data that we generated a number of years ago. I'm looking at bile acid synthesis changes following a toxic drug exposure. We can look at the proteomic changes that occur in the cells as well. And again, a wide variety of peer-reviewed publications in this space. And then finally, we can use this system for disease modeling, and we have a very well-established and very detailed model of non-alcoholic fatty liver disease and a number of other liver diseases such as viral hepatitis. So here we induce a liver disease state over a period of time with a presence of fats and sugars, and we create all the endpoints that's seen in human disease. So we have steatosis, fat loading, we have fibrosis, we have inflammation, and we have a wide variety of endpoints again that we can assess and quantitatively determine how the microtissues change as a result of these stimuli. The transcriptomic profile of the model again matches the human profile. So we put the transcriptomic profile into an online portal. It thinks we're looking at a NASH biopsy, and it much better correlates with human samples than standard murine models that are more typically used. We can use this to screen molecules. I've done so with many parties now, looking at antifibrotics, antisteatotics, etc., to understand how they behave. And again, same story. We have a wide variety of peer-reviewed publications in this space, really demonstrating, in particular, also a number of publications looking at target ID, looking for novel targets for this disease. So to round off, I should also mention that this liver MPS has been evaluated by the regulator. So we had a publication with the group within CEDAR at FDA last year. We were the first MPS developer to have this type of publication. And they demonstrate a lot of the things I already showed you. So the metabolic capacity in this system is significantly higher than standard spheroidal or sandwich cultures, and it's much further extended. So here we go out to 28 days of metabolic activity. This is CYP3A4 activity. It's highly reproducible, which is clearly very important, and can be used for both drug metabolism and toxicological applications. And you see some of the tox data here of a couple of different compounds. So with that, I'm pretty much out of time, so I'm going to stop. But hopefully I've shown you just
you've got 1 minute.
Very good.
1 minute.
How we've used our liver MPS in a number of different spaces. We have a really broad variety of peer-reviewed publications in this area, and we think we have one of the most advanced liver models available. But it's also very easy to use, easy to set up for parties to allow them to really incorporate it into their workflows very straightforwardly. There's lots more information available. Please reach out to us. And I should say that I know the original technology for this was developed by Linda Griffith at MIT almost a decade ago now. So we still work very closely with Linda and her team, and the work that we've done with them has been supported by the work with other universities across time. So with that, I will stop.
Thank you. You're about spot on. We started about 3 minutes late. You've got one question which we might find easier to ask in the group, but based on images you've shown, looks like your platform includes media recirculation, not unidirectional flow. Is this correct? If so, I wonder how different your model than 3D spheroid models. I think that's probably quite a big question, but are you able to answer that very briefly?
So I guess the brief answer is yes, we have recirculating flow, not single pass. So from a practical perspective, that means we have to replenish the media on a regular basis, but the system has the capacity to actually run for quite a long period of time if we want to. So we can leave the system untouched for a week if we need to for a particular application. So there's quite a lot of flexibility there.
I suspect similar questions will come to others, and we'll move over to. Thanks very much there, and we'll move over to Draper.
Okay, can everyone see my presentation?
All good.
Okay, awesome. All right, well, thank you so much to the organizers for putting together this great showcase of liver technology, and we're very happy to be part of it. So Draper is a nonprofit engineering firm. We've actually been involved in this space for some time, for several decades. Participated in some of the first bilayer MPS models in the vascular space. This is done with Jeff Bornstein, who's also still overseeing many of our organ-on-chip programs. More recently, we developed a tissue with Teresa Woodruff at Northwestern, female reproductive tract. And so we're fairly new to the liver space because in the last 5 years, we've specifically focused on developing platforms for organ-on-chip culture that are more amenable to integration into pharma pipeline and drug discovery and testing, specifically with the result is scalability. So in that regard, we developed Predict96 is our flagship platform. It's a high throughput platform. Some of the highlights, the systems that we've have collaborations with 12 of the top 15 pharmas. Most of them have the system and the tissue models integrated within within in-house. More recently, we developed an airway model on this platform that is, to my knowledge, one of the first and still one of the only organ-on-chip models to show robust SARS-CoV-2 infection. So generally, what's governed our design is be able to cram as many knobs as possible in an in vitro platform to control tissue structure and function in a precisely controlled way at scale, and then integration of sensors and real-time data collection to kind of reduce the hands-on necessary needs for interrogating the tissue and kind of in a way releasing the organ-on-chip bang for your buck. So Predict96 platform, which we develop our liver models on, consists of a tissue culture plate and a sterile lid. So the tissue culture plate is 96 completely independent organs. As you see in the top left, they're in a bilayer format. You can sort of think of this as a souped up transwell. That's a much more optically optimized, better cell volume ratios, and more importantly, contains recirculating fluidic flow independently controlled in the top and bottom channel. So it's controlled by this fluidic lid, which is just a little bit bigger than the culture plate itself, which has 192 micropumps integrated into the lid. So we can get independent control, one pump for each channel of each device. We've also integrated different sensors to interrogate the tissue in a more facile way. We can do flow at 0 to 10 dynes per centimeter squared, and we can program remotely both the sensor data acquisition as well as the flow. So as mentioned before, we designed this footprint to be implementably into both small therapeutic and large therapeutic pipelines. Our footprint is very small, so we can fit 2,000 to 3,000 organs in an incubator if we so wanted to. We can use standard tools to facilitate maintenance of the tissue and seeding and generation, including micropipettes of the liquid handling systems, all of which we use in-house to kind of reduce the hand touch system touch points of our generating our models. We've integrated tear and O2 sensors to interrogate the tissue in a more trofaccal and granular way. We'd be able to use lots of proteomics, omics kind of transcriptomics and proteomics applications of our data assimilation, as well as rapid high content imaging. As you can see in the bottom right, this is a Phoenix image of the 96 devices, 5 colors, 40x, 20 different stacks in about an hour. So it's amenable for all these high data systems. And for almost all of the transcriptomics, to say single cell RNA sequencing, we don't have to pool samples for most of our tissue models. So we get true 96 device throughput. All right, so I've told you a little bit about the system we developed, the platform. I'm going to tell you more about the liver biology that we've generated in the platform. So again, we have a number of knobs we can tune, and we can take advantage of the throughput to combinatorially assess these knobs to create the most optimal in vivo physiological environment for our tissue. So in the liver regard, what we've done is we've looked at a number of different knobs, so to speak, of extracellular matrix, membrane properties, oxygen transport, fluidic flow to recapitulate mass transport regimes that are more sinusoidal-like, et cetera, in a combinatorial way. And we've optimized the parameters surrounding this liver biology to create more sustained hepatocyte, stellate, and Kupffer cell tissues. So as an example of this, in the bottom left, you can see a graph of flow rate versus theoretical platelet number. So we've used empirical data as well as modeling to come up with a fluid flow regime that recapitulates the in vivo sinusoidal mass transport of small proteins, which is assigned with this theoretical platelet number. And we use this for many of our applications in liver space. The TriCell model that we have includes typically hepatocytes in the top channel with a collagen overlay, co-cultured with stellate cells, and Kupffer cells and indirect co-culture on the bottom with the fluidic flow. So some of the really nice design features of the system is that we can, if we wanted to, concentrate the cell volume ratio at a much higher level, closer to in vivo. And so what that does is it works twofold: to concentrate the secreted factors and provide a higher dynamic range of detection, as well as augments the cell health metrics. So you can see in the top and bottom albumin and CYP3A4 activity, we get higher levels that we can detect, but also on a per cell basis, we get augmented tissue health metrics. And this is independent of the fluid flow. So when we actually add the fluid flow that we talked about before, we get an even more augmented cell health metrics. So this is a panel of health metrics: urea, CYP3A4, albumin, hypontrypsin, VEGF, HGF. We get levels that are acceptable for in vivo physiological standards. And you can see in the red the flow versus the black static, that we get 2 to 3 times higher levels when we add the fluid flow. And then that those health metrics are sustained for longer periods of time. So typically we can get about 3 to 4 week cultures before we start seeing significant drop off in these health metrics. All right, so we've also been able to correlate some of this fluid flow driving hepatocyte mature phenotype. So on the left, we have a collagen 1 probe that we can actually measure in real time the collagen production that's produced. By day 7, we get robust collagen production of the endogenous hepatocytes. And moreover, the flow actually augments this. And at the same time point, we also see evidence of biocaniculi, strong adherent expression. This is evidence of tissue stabilization and polarization. We can further quantify this easily using visual methods or endopermeability assays, which we've done in the past. Okay, and another really unique feature that's related to the liver model that kind of unleashes the utility of this model is we have a pressure seeding system, which allows us to culture previously hard to culture, impossible to culture cell types. So this is really important for liver, where these are post-mitotic differentiated cells which don't proliferate. So you really need, they're really sensitive to the quality of cells. Many lots you can't use in a traditional sense. So we've taken lots that our collaborators haven't been able to use. This is a transporter qualified lot, which is terrible for seeding passively, and we're able to get them to culture in our tissue. The coverage is not as good as a healthy tissue, but we're still able to get sustained tissue for up to about 3 weeks. You can see on the right, CYP activity at high levels, also augmented by flow. On the bottom, we have evidence of NASH patient cells we get from our collaborators that, again, very hard to culture for various reasons in vitro on a tissue culture plate, but we can culture these in our systems for about 10 days before we see drop off in sort of the functionality. Okay, so obviously an important part of organ-on-chip in general is to be able to create complex systems, but also deconvolve the variables that surround disease etiology. So in this case, the cell contributions that affect disease response. So this is sort of a proof of principle demonstration, primary hepatocytes and the top channel and primary Cooper cells in the bottom channel. So we simulated liver damage by activating primary Cooper cells through LPS exposure. This does not affect primary hepatocytes alone, but when you add the Cooper cells, you get both negative and positive acute responses. So negatively, you get a reduction in CYP activity, but an increase over time in alpha-1 antitrypsin and VEGF. So it allows us to easily probe how specific cellular components can affect parenchymal function. We've done a lot more studies with proteomics and transcriptomics to kind of probe in this mechanism of action in more detail. Unfortunately, we can't show that data in a non-NDA CDA setting, but we'd be happy to talk more about that if you're interested offline. So similarly, we can pinpoint cell crosstalk amongst stellate cells and primary cells. Hepatocytes, you can see on the left, we have a co-culture model as well. When we actually add the stellate cells, we get augmented CYP3A4 activity. And then on the right graph, when we add a TGF-beta, which is of course a driver of liver inflammation in vivo, myofibroblast, EMT, as well as dedifferentiation of hepatocytes, we get a reduction in the CYP3A4 activity, which is unsurprising and what we expect from in vivo physiology. On the right, when we add TGF-beta again, we get this reduction in CYP4 activity. When we add LPS, we get no response. But when we add Cooper cells in this tri-cell model, just LPS alone, without the TGF-beta driver, will also starkly reduce the hepatocyte metabolic function. And we've actually correlated this in more studies to correlate with the activated Cooper cells as becoming a TGF-beta and other pro-inflammatory cell source, which again is very important for early liver disease and damage and disease progression. So again, collectively, this model shows the ability to do the crosstalk and understand crosstalk and how the cells respond to different environments. All right, so finally, we demonstrated steatotic induction in this multi-lever cell chip. This is kind of using traditional methods of chronic dosing of unsaturated saturated fatty acids to induce a steatotic state in otherwise, quote, healthy tissue, and found a biphasic response here where low levels augment metabolic function and high levels rapidly mitigate this function. This is actually reflects some clinical data that can show you that where this kind of biphasic response in human diets can also affect liver health. So there's ways to endogenously create these disease models with healthy tissue, as well as in our system be able to use actually disease patient cells to recapitulate some disease etiology. All right, so in summary, we developed this liver system with this Predict96 high throughput scale, allows onboard pumping, recirculating flow, maximum tunability of the tissue for different disease applications, easy to use standard footprint, and that's automatable and implementable in a pipeline. Thermoplastic materials that reduce drug absorption, great optical properties. So pretty much any assay you want to use, you can use in this assay. And we've tried to capture a number of in vivo responses in disease etiologies. Unfortunately, most I can't. The big data I can't show you in this setting, but in a private setting, likely can show most of that data, and hopefully most of it will be published soon as well. So that's it for this talk. Happy to take any questions.
Awesome. And I'm back to help coordinate any questions. I don't see any questions at the moment. Does anyone have any questions for Draper at this time? We'll give it just a little bit, and if not, then we'll go ahead and direct people to ask questions during the roundtable. Okay, so with that, we can go ahead and move on to emulate. Awesome. Hi, Ben.
Hello, thank you so much.
And again, to all of our participants, feel free to use the chat to ask Q&A anytime, and we can always get to it during the roundtable, and I'll turn it over to Ben.
Okay, thank you so much. So my name is Ben Swenner. I'm a scientist at Emulate. I'm stepping in for Danny Levner, our Chief Technology Officer, as he had a last-minute family emergency that came up that sadly prevented him from being here today. So I'm going to be walking you through how being able to improve predictive validity using Emulate's liver chip. So as many are familiar with the human story of translational failures, that we can see that drug-induced liver injury remains just a leading patient safety concern, even presently so. We can see here on the left that, with these case studies that are very widely known and well studied, but even as recently as January of this year with Pfizer Ionis discontinuing buprenorphine due to elevated transaminases as a cardiovascular disease therapy. And so when we look at this and we see that current workflows for in vitro and animal models are inadequate, and which is of course why we're all here today and want to be able to discuss this. And so what we've set out to do is to be able to build off of this, where we can see that outlined from IQ and being able to set out these criteria for a liver MPS system. We then use this criteria to then be able to qualify our human liver chip for predictive toxicity. So in this case, what we wanted to be able to do is be able to fully, kind of qualify based upon those guidelines that in that previous publication that was outlined a few years ago. And so with this, what we can see is that we were able to evaluate 18 out of the 20 drugs set out by IQ MPS. We were able to then be able to look at those major mechanisms of drug-induced liver injury that they were represented on our chip, and then as well that all drugs that were tested in blind concentrations and that they were adjusted for protein in chip media. Additionally, we were able to use concentration range tested up to 300 times the free human Cmax that was obtained, that was readily available in literature. And again, today I'll be giving you a brief overview of this and this work that we were able to do. But if we want to be able to have a deeper dive, feel free to reach out to us and we'll be able to happily talk more in depth about this. So our study design is that we were able to use our human liver chip. So if we have a 2-channel system separated by a porous membrane, and that top channel is where our primary hepatocytes would be. And then that bottom channel is where we have our non-parenchymal cells, in this case being the Kupffer cells, stellate cells, and liver sinusoidal and endothelial cells. And so here what we're able to then see is that we're able to then have flow. We're able to then adjust for what we were able to need. So we were able to, depending on solubility, and of the compounds that we're able to then adjust and differ and optimize the flow rates for each of those compounds specifically. All cells used in these studies were human primary derived, and as well as we used 2 different hepatocyte donors throughout testing. So the study design and how it looked like was that we initially have, days of our prepping. So we were able to then activate coat our chips. Then we then seed with the hepatocytes and then followed by the non-parenchymal cells. We let those stabilize then for 5 days. And what you'll see here is then from days 5 to day 12 is when we challenge the chips with our drug testing and doing endpoint analysis. So we did a 780 chip experiment. So one of the largest, chip studies that's been kind of run today that I've seen in publication. And we broke this down into 4 different cycles. So cycle 1 and 2 tested 25 out of the 27 drugs that we evaluated. And cycle 3 tested 14 of those in the second donor. And then cycle 4 was testing 6 drugs in donor 1 and another 4 drugs in donor 2. RNA sequencing was performed on these chips, and it was performed on both days 3 and 5 during cycle 3. And so what we can see is that also when I talk about data going forward, you'll see that when we talk about days on, looking at endpoints on days either 1, 3, and 7, that is going to be 1, 3, and 7 of the drug dosage, not necessarily on how long these cells have been directly on the chip itself. So as far as terminal endpoints, we were able to do live stain imaging, so we can do caspase 3 and 7 and TMRM. To be able to look for apoptosis and toxicity, but then we also can do MRP2 and nuke flu as well. we go. So what we were able to find is that our human liver chips will, for one, being able to qualify them as being able to exhibit typical hepatic phenotypic markers. So we can see under bright field microscopy there on the left, in that top right, sorry, in that top left rather, would be our primary hepatocytes, and then with our non-parenchymal cells there in that bottom left. Then when we look at under immunofluorescent staining, we can see the staining of the Kupffer cells with CD68, Desmond for the stellate cells, CD31 for those sinusoidal endothelial cells, and then MRP2 for the bile caniculi. Additionally, we can do TEM microscopy, and we're able to look at those bile caniculi then directly on chip. So we can see some really nice, beautiful images there. But in addition to that, we can also look at those interfaces between the Kupffer cells and the endothelial cells directly there that's on chip. We can really see how they interface and really have that close proximity to one another. So for baselines as well, for wanting to see, how are we, defining that we have healthy hepatocellular functionality, we determined that we would want to be able to see that we had, albumin production and urea. And so we have that here, that we have those outlined again on days 1, 3, and 7. And this is after having drug being dosed. And then we can see this across both different donors. And the dotted lines here is for reference from what we found in literature for having a physiological range of what human liver would be able to secrete for albumin and for urea. So we can see that we use these as a good baseline, that we do have functioning and active hepatocytes, and that we're good to go and maintaining that for our chips. But in addition to that, we wanted to be able to look at being able to distinguish between toxic and lesser toxic structural analogs. And so we can see that when we look at albumin, and in this case, what we are looking at is trivofloxacin and levofloxacin. And we can see that, after looking at a multiplier of human Cmax, we can see that, starting right around, that 20, 30 times the Cmax, we start to really see that tapering off for the trivofloxacin, that more toxic compound that would be found comparatively to its lesser toxic compound levofloxacin. Additionally, we see this correlate with ALT secretion, and we can see that we're looking at ALT that's directly found on chip through our effluent. And additionally, we can look at our morphology as well. So we use this, we have a way of being able to score our morphology based upon the presence or toxicity of the cells, and we can grade those accordingly, but both by bright field microscopy, but as well followed up with immunofluorescence. So looking at some terminal immunofluorescent imaging. So when we look at, we're looking here at caspase 3 and 7, we can see that trivofloxacin, again at that 100x, we really see that upregulation of that apoptosis, seeing that we're having that, more severe response in response to that compound, or to that drug rather. And then also with levofloxacin, we're seeing that we don't actually see that. So we're not seeing this all the way up to 45 times its concentration. And there are differences between 45 and 100 just due to the solubility of levofloxacin that prevented us from being able to go higher Cmax. But we were overall able to then see that with the potential of the apoptotic cell death that we're seeing in response to trivofloxacin. So now when we were looking at being able to look at this for the matched pairs, we're looking at 7 different matched pairs here. We can see that when we look at how they would be able to show with the more toxic drug analog, we can see that we have downregulation of albumin across all 7. We see that we have in all but 1, we have upregulation of ALT, and as well as that when we look at the morphology score, that we're seeing a higher score, in this case indicative of higher toxicity, that we're seeing that is also then present in 6 out of the 7 compounds there. Comparatively to the lesser toxic drugs, we can then see there with albumin levels that they were non-existent, that they were at baseline. We didn't see any effects there with downregulation with this one compound here. So what we're able to then say is that we're able to see that we have, for 7 out of 7, we're able to predict, excuse me, that we're able to differentiate between the toxic versus less toxic compounds proposed by the IQ MPS guidelines. So when we then look through the quantitative performance of the liver chip, so when we have this and we're kind of setting up in this grid format to be able to see that if it showed up as DILI positive in the clinic or versus DILI negative. And then if the model was positive versus the model being negative, we have that true positive, false positive, false negative and true negative there. We can see that the animal models clearly had missed, at a 100% rate of being able to predict DILI. That was found in these patients, and where patient concern is just so great, and that's our main focus here, and we're wanting to be able to improve that. We can see that, again, this just demonstrates what we all have already known. Spheroids definitely bumped that up to 47% specificity, but still in 100% specificity, 47% sensitivity, sorry. And we can see then, but they were still seeing a multiple of them that we were able to see as false negative, those 10 there. Then when we moved on to chips, and this is all corrected for protein concentration or protein binding, we can see that on donor 1 that we were able to get a rate of 77% sensitivity on these 27 drugs. And then also on donor 2, we were able to have 73% specificity, where we missed 4 here and 5 out of these respectively. When we combine them and we're able to pull that data together and we look overall across those 18 drugs that were run on both donors, we can see that we had an 87% sensitivity rate that we can then further be able to extrapolate and build this up. That we really want to be able to have that as we're able to have more and more donors present on chip, that just allows for that donor to donor variability that would oftentimes, or that would always be present in clinical trials. We're able to then see that improvement in sensitivity there. So, and as I said, if we're wanting to be able to set up a deeper review of this study, please reach out to us. Happy to do so. I want to just say and acknowledge all those at the team that were able to go over, that did all this hard work and be able to analyze this, as well as thank you to those that were being able to review this manuscript currently, Dr. Jack Scannell for his advisement as well, and of course Dr. Ingber, who is our scientific founder, and all the work and advisement he has given us. Thank you.
Awesome, thank you, Ben. Does anyone have any questions for Emulate? We'll let it go for just a minute to see if anyone chimes in. As a reminder, you can always put questions in the chat at any time, and we can always answer them during the roundtable if we don't get them today or right now. Okay, so seeing none, we will go ahead and move on to Hesperos. Thank you, Ben. Oh, it is Jay.
Hey, how you doing, Megan?
Good to see you. Yeah, someone else had logged in for Jay earlier, so good to see you.
Yeah, I had something else I had to do between 9 and 10, so, but I'm here.
No worries. Glad you can join us now. So if you want to go ahead and share your screen. Should be the little arrow to the left of the red leave button.
How's that?
Looks good if you just want to move to the presenter mode. And then I will jump off and give you the mic. Awesome. It looks like you can, if you want to back up to your first slide or start here, either way is good.
No, this is fine.
Perfect.
So I'll give a quick overview of Hesperos. It was founded by Mike Schuler and I, based upon about 20 patents from Cornell and UCF. Mike and I have been working in this area for about 25 years each. We're a CRO. We do not offer our systems for sale. We won the Lush Prize in 2015. We've used SBIRs to bridge the valley of death, and we're quite healthy right now. We moved into a 14,000 square foot facility down here in Orlando. We've got about 35 people at the company. We're really focused on increasing efficacy, lowering costs, and decreasing timeline for drug discovery, as we're providing new solutions for rare diseases. As a matter of fact, we had the first MPS IND filed in December of 2020 for a rare disease CIDP with Sanofi, which basically was allowed to proceed to clinical trial based upon the phase 1 safety data they had for another indication. And then our data was the only efficacy data they had for the new indication. Again, that's currently in a clinical trial. It's a very busy slide, but just wanted to kind of show, you know, where this started was Mike had an idea about 25, 30 years ago that you take a PBPK model and use those equations normally used for predicting concentration in phase 1, and a subset of those to design a multi-organ system, look at metabolism. We've now advanced that to the point where we can add in all kinds of different chips into the system. This is an example of a 5-organ system we're using for looking at opioid overdose and recovery, as well as off-target tox. But a key point to this is we use what is called a pumpless system. So we use gravity to pump it. So we get rid of all the tubing, all the pumps, all the bubbles. But most importantly, especially for this presentation, makes it a low volume system. So we can see the effect of both metabolite and parent in the same system on a compound, then its effect on other organs. The other thing that we have developed is this idea of clinically relevant functional readouts. I've been doing this for about 25 years. And so again, the idea is for Hesperos is that when you walk into a doctor's office, he doesn't immediately stick a needle in your arm to check biomarkers. So how are you walking? How are you talking? He listens to your heart. He checks your reflexes. These are clinically relevant functional readouts, things that we can reproduce in our systems by being able to look at muscle contraction, electrical activity for neurons and cardiac cells, neuromuscular junction formation, barrier integrity. Now let's look non-invasively for acute, but most important for chronic monitoring in these systems, and then mechanistic determination of tox and for target identification. And again, just kind of goes through looking at microelectrodes with neuronal systems, looking at pattern cardiomyocytes over electrodes, looking at mechanical, basically taking the cells and putting on thin cantilevers to be able to look at force with skeletal muscle and cardiac. We can look at biomarkers, in this case liver, but also barrier formation in these systems. So we have developed a serum-free media that allows hepatocytes to go in 2D and 3D out for 28 days in the system. We maintain good albumin, urea function, but also CYP activity for basal function and also induced. This was published in Biotechnology Progress. We'll be able to take these things out for 28 days and hepatocytes only. In these systems, we do get biocaniculate formation. You can see in those systems after 28 days. Now, what we can do with this system is we can then selectively add in Kupffer and stellate cells and see what the effect is of drugs on the different combinations, where we can look at the effect of the imatinib and diclofenac on the system and basically show that imatinib, there basically is no effect on whatever combination. But as you can see, as you can add in the Kupffer and the stellate cells, that there is a big difference between liver only and the combination of cells. And we can see that, okay, in our system because we can selectively add in each cell into the serum-free media. We can also look at stellate cell activation, look at fibrosis in the system by treating it with TGF-beta and CTGF. And you can clearly see from the control and then going to the decrease, we can also see the effect of fibrosis in the system. We can also, again, add this and look at multi-organ systems. Again, our real focus is to look at multi-organ systems where we can custom build what people are interested in, where if you're interested in this case, fat and liver, we can actually create a white adipose tissue model with liver. We can design it so we have the proper flow rates, the proper dimensions. In this particular case, we characterize hepatocytes. We're trying to create looking at a liver NASH model, an FLD. And this is the initial experiments to try and be able to do that. If we see hepatocytes by themselves, we treat it with the inflammation inducer, which is TNF alpha. We don't see much effect. We add it with lipids and also TNF alpha and lipids. We really see some effect in lipids with healthy and diabetic media and hepatocytes by themselves for looking at lipid accumulation. We then looked at adipocytes in monoculture and showing that the inflammation inducer was able to decrease the lipid accumulation the lipids were increased to some degree, and there's a difference between the healthy and the diabetic media. And again, the TNF alpha basically attenuated that. However, when you then add the two together, you can clearly see the addition of the TNF alpha causes a accumulation, okay, in the liver, okay, and actually you see the lipids, you see the combination thereof. So what is happening is the inflammation in the white adipose tissue is causing lipid accumulation into the liver. And so it's only that drug, that multi-organ system linking it together and having a communication that we're able to see this effect of the white adipose tissue on the liver. And be able to see that the CYP activity was decreased by the inflammation, okay, of signals coming from the fat. You can see that the adiponectin, okay, is decreased, okay, with the TNF alpha. And see it's increased with the diabetic media. And again, you can see the two, it's decreased, adiponectin with the inflammation marker. You can see that the leptin, okay, in the diabetic media increases dramatically, okay, in the presence of the inflammation, okay, and again, that is maintained as well. And this was published in Nature Scientific Reports. Again, showing that this cross communication between organs is extremely important, which is one of the main focuses of Hesperos. Going on that same theme, again, we do look at biomarkers in terms of the liver viability. What we really try to do is look at function in a collaboration with AstraZeneca. We wanted to look at terifenadine, fexofenadine couple in the same system. So we combined this cardiac electrical activity and force, okay, with liver in a recirculating system, and be able to show that terifenadine in a cardiac only cause a QT prolongation, which is the reason it was pulled off the market. Its metabolite, fexofenadine, which is now Allegra, okay, does not have that cardiotoxicity. You can see that it's shifted when the liver is present. Again, the conduction velocity, okay, was not affected with a heart-liver system, whereas in heart only, you see a toxicity about 33 micromolar in the system. But again, a key point in terms of these clinically relevant functional readouts, we do this without cell death, okay? Because if you're getting cell death in your clinical trial, you have a large problem. So what we try to do is predict what would actually happen by decreasing the functional aspects of it, again, without looking at cell death. We can look at viability changes, but again, this is really what you're not looking for in a clinical trial. Now, and this is really important because what we can do, we can model these systems. We can actually then grid them up. We can look at the interaction of compounds absorbing, like fexofenadine does not absorb much. Terifenadine absorbs quite a bit. We can look at mixing in the system. We can look at the loss, but we can then generate the area under the curve versus the as treated. Most papers, what they do is this is the concentration we administer to the system. They don't really monitor for area under the curve, but being able to look at area under the curve, we can then model the in the system chronically for fexofenadine, the metabolite, and the parent terifenadine. We can then do HPLC MS monitoring to show that we actually have pretty good agreement. And we can use this to create a PKP models of our system where we can look at PK, looking at counting for any absorption, the metabolism, and again, the due to the PK of the metabolism. We can then predict the PD relationship for the interaction with the receptors on the outside of the cardiomyocytes, but we can also look at the internalization, in this case, looking how the field potential dependence on the effect of the hERG channel, be able to create the PD model for that as well. And then we use that data to be able to predict data that for guinea pig, dog, and non-human primate found very good agreement with this. And this was published in Nature Scientific Reports. And again, showing that what we really are focusing on is the function, the ability of the liver in the multi-organ system to be able to affect the other organs around it, where it's functioning as in creating the metabolites that you'd want to be able to see in the system. Again, we can put this into multi-organ systems. We published 4 organ systems to be able to evaluate compounds. It's published in Nature Scientific Reports. We've taken this out for 28 days. It's published in Advanced Functional Materials. And again, just one figure from the paper, basically showing that we can maintain the hepatocytes for 28 days in a recirculating system, and basically showing that the compared to static, the static falls off as you'd expect, whereas the under flow with just the hepatocytes, you see there's some variability, but in the presence of the organs, you see a very stable system for 28 days. And these lines here are different literature values for the albumin production. This is for urea production. And again, monitoring the CYP activity in the systems for 28 days, showing that very, very stable, and the presence of the other organs actually helps stabilize the liver for that period of time. We're also able to get circulating immune cells and see their interaction. In this case, we were able to reproduce the functions of the innate immune system by being able to predict the M1 and the M2 phenotypes, and again, in the presence of a liver, and be able to look at the fact of the cytokine storm, where it attacked all the different organs, and then selectively damaging one of the organs caused the M2 or restorative phenotype. And this was published in Advanced Science. And lastly, just showing that we can integrate barrier tissues with these systems. We worked with L'Oreal for years. We're able to put skin into the systems. I showed you earlier, we can do kidney only as a tox target. We don't have an elimination model. We can add in blood-brain barrier and also GI tract in the system as well. Mike Schuller has worked on these models for years. These are easily integratable into our multi-organ systems. So what I really wanted to do is to show you that we have very competent, very functional liver models, but what we can do, we can integrate them in the same serum-free media with many other organ types. So that's really what our focus is custom building multi-organ systems for whatever problems or issues you might have or disease models you're trying to do. So this is the team at both Hesperos and UCF in front of our building. I'd like to thank them. I'd like to thank you for your attention, and I'm ready for questions.
Awesome. Does anyone have any questions for James at this minute? Okay, I think we're going to be saving some questions for the round table. So thank you, James, for that overview. And we will move on to our last presentation today, which is from InSphero. So go ahead, Sue, if you want to share your screen.
Sure.
And your camera is currently off if you want to turn it on and show your beautiful face.
Okay, give me 1 second here.
Yep, no worries. We can see your screen.
Okay, wonderful. I think we're all set. Thank you so much. Thank you to the NA3Rs Collaborative and to the IQ MPS Initiative. It's quite an honor for InSphero to be represented within today's liver MPS workshop. And since the great presentations before me already highlighted some of the limitations of animal research and the need for better in vitro human models for predicting liver toxicity and disease, I'll jump right into the background of my company, InSphero, how we're also working to produce human cell-based models which represent the clinic. So here we are. 1 second. It is not waking up. So the entire foundation of InSphero's patented technology started about 12 years ago with scaffold-free self-aggregation of cells known as hanging drop technology. As you can see here, hanging drop occurs when you allow cells to self-aggregate within a hanging droplet of scaffold-free media. So no animal containing scaffold, such as rat collagen or mouse-derived matrigel. After a few days in this droplet of media, these cells naturally find their way together and reestablish tight cell-cell connections. Then additional media is added to drop the microtissues down into our patented ULA-coated Acora plates. So this approach can be done with numerous cell types, including cell lines, primary cells, iPSC-derived cells, and the complexity can also be adjusted. For example, monoculture of one cell type versus co-culture of multiple cell types representing a particular organ, with the goal, of course, to get that much closer to in vivo human. So since the topic of this workshop is all about liver, I'll focus in specifically on our 3D Insight liver microtissues. So our human liver microtissues are offered in several options depending on the research question being asked, but the two main models contain cryopreserved human hepatocytes, Kupffer cells, and endothelial cells for toxicology studies. And then for liver disease research, we can also add in stellate cells. As I mentioned already, there are no artificial matrices added to support the culturing of these microtissues. They actually create their own matrices and in a more physiological manner. So it was critical when we validated our liver models that the essential liver functions were maintained. This includes drug metabolic activities, as it's known that these important hepatocyte functions decrease significantly in simpler in vitro cultures. We can measure bile acid secretion and further measure drugs' effects on this secretion. And this is incredibly important because that means the cells have reestablished the critical cell-cell connections necessary for bile networks to be reestablished, and that efflux transporters are properly aligned. And as we've already heard here today, albumin production and secretion is a critical hepatocyte function in vivo. So we wanted to ensure that this function was retained in our culture. In fact, you'll typically see a decrease in albumin secretion prior to other measures of cell health and viability, so it can be quite an insightful endpoint in studies. And another major distinction, which was also highlighted earlier, is the ability to culture cells long enough to recreate conditions of toxicity or to mirror and then treat a disease. So diseases like NASH take years to manifest in humans. So the tricky goal of in vitro models is to recreate unhealthy cells that reflect a disease without killing them, but then they can still be recovered by standard of care or clinical compounds. And this takes incredible R&D efforts, which I believe our company has really tried to work towards. So with the hanging drop technology comes the ability to create numerous plates, easily several thousand individual yet nearly identical microtissues in a single production. Each production undergoes stringent QC, as you can see in the minimal batch-to-batch graph here in the middle of the slide, where ATP and diameter must fall within a very tight criteria. We have an instrument which can take a quick snapshot of a plate and quickly measure and assess the diameter of each microtissue, as you can see here in the heat map in the lower left-hand corner as well. So with the scalability of this many microtissues per production, combined with 384-well plates and optimized assays for these little microtissues, we can accomplish a remarkable amount of screening. But then the question is, what are these microtissues used for? So liver toxicity, of course, is a major application, as we've heard earlier, for in vitro models. While it might seem inconceivable that these little microtissues could possibly represent human liver, you have to keep in mind that they contain all the relevant human cell types. They are a primary human cell origin. They retain in vivo-like function, and they remain healthy for many weeks unless you treat them with something toxic. And you can see here on the left-hand side all the different types of cellular DILI key events that can be recapitulated and measured. These can be measured through a variety of different manners, often from the same single microtissue. There are biochemical readouts that can be multiplexed, for example, ATP from cell lysates and secreted ALT from supernatants. And histopathology is something we regularly do, but because you have to pool so many of them, frankly, we've invested much more recently into confocal imaging because only a single microtissue is required. It's easier to actually quantify the results, and there's the possibility for determining co-localization. For example, if you want to tie an event to a specific type of cell within that microtissue. And last point I'd like to make on the slide is that there's been great advances in omics within liver toxicity and disease. You've already heard some of this. And one thing we're doing now regularly is utilizing TempoSeq technology from company BioSpider for analyzing many thousands of genes, all from a single microtissue. Now, of course, we still use 6 biological replicates, but it's still pretty astonishing what you can measure from less than 3,000 cells in a microtissue. So one thing that makes InSphero stand out is that our model is highly scalable and offered as a service or as assay-ready plates that can be shipped to customers. And so for companies that want to perform DILI hazards identification screening. Now for translation to man, we really want to help bridge that gap from the in vivo tox animal to that of human in the hopes that in vivo animal testing will not be as dependent upon in the future. We actually have a major consortium effort going on right now, including several major pharma. So for developmental drugs, which already show signs of toxicity, there needs to have better understanding of the mechanism of this toxicity. And we can achieve this by investigating different types of DILI events, as shown in the previous slide, for example, reactive metabolites, disease risk factors, mitochondrial tox, and so on. And lastly, we have been asked to support situations where a clinical drug has caused hepatotoxicity. And if this is believed to be an idiosyncratic effect, then we have the ability to incorporate human donors with specific genetic backgrounds, for example, specific HLAs or genetic SNPs into our model. So as you can imagine, all of us humans are very different. And so what we put into our humans models will, of course, affect the research that comes out. Now, I'd like to quickly show how we built upon our hepatotoxicity model into an even more complex NAFLD-NASH model. Of course, the first thing we accomplished was the arduous task of incorporating primary human stellate cells, which are not already activated. Then we wanted to faithfully recapitulate the stimuli that's represented in the clinic. And instead of 10 years, we wanted to do it in vitro in 10 days. So this stimuli means increased sugars, free fatty acids, and a pulse of inflammatory stimuli that represents the leaky gut that occurs in NASH. And because genetic SNPs are becoming such more relevant in NASH research, we can actually replace out our standard cryopreserved pool donor lot with a donor with a specific SNP of interest, for example, and easily incorporate that individual donor into this model. So we can use all the same types of readouts that we already developed for hepatotoxicity studies, but now also add endpoints that are more specific to NASH initiation and progression. There are so many different pathways researchers are investigating in NASH, and so I should point out not all of them directly target the liver. And so a liver-only model like ours would not be appropriate for their research. However, our liver model does nicely show de novo lipogenesis and the development of steatosis, that first stage of NAFLD. Then we can clearly see increases in numerous cytokines and chemokines associated with the inflammatory stage of NASH. And then lastly, perhaps most importantly, we can measure significant NASH-induced fibrotic signals after NASH stimuli in 10 days. And using tool compounds or antibodies, we can block these signals. We are in the process of comparing dozens of clinical human samples with serous red staining slices of our microtissues to ensure that we are achieving certain stages of NASH relevant fibrosis. And we use machine learning based image analyses for this. So in very brief summary, and we recently published in this Nature Reviews, our readouts for NASH research include secreted factors, lytic assays, microscopy, and gene expression. And just to give a visual of what our healthy lean versus NASH treated microtissues look like, here are some H&E stainings after 10 days on the left-hand side. And in the middle, we can measure intracellular triglycerides using Promega's TG kit. And then images taken on our Yokogawa Cell Voyager CQ1 high content analysis system, where Nile red staining can also be quantified. So while I'm sharing focal images, I didn't know if anyone else would bring this up, but I wanted to quickly sidebar to the fact that there's been a movement in the field towards RNA drug modalities. And this makes sense for treating liver disease, as these drugs will inevitably go to the liver anyhow. But to make drugs more specifically target hepatocytes, researchers will conjugate their ASO or siRNA to GalNAc, which requires a hepatocyte-specific receptor, ASGPR1, for entry. The trouble researchers are finding with simpler models I've heard, for example, 2D plated hepatocytes, is that ASGPR expression essentially disappears, either at the gene level or it internalizes when plating in 2D. So what we've been really excited to show is that ASGPR1 expression is indeed retained and properly expressed when cultured in 3D, as shown here in our lean and NASH microtissues at day 10. And in some of our client studies, we can easily confirm that there is much greater entry of GalNAc conjugated drugs compared to their naked versions as measured by gene knockdown. So now on very quickly to how we're building upon our microtissue technology towards body on a chip. I've been focusing today on our liver models, but of course the liver influences so many other systems and organs we've already heard today within the body of vivo. And so wouldn't it be wonderful if we can try to recreate some of these connections? These are examples of different microtissues that we can produce from different organs. And now we are trying to find ways to have them interact with each other and offer solutions to better address very specific scientific questions. So the overall idea is that we still use our fundamental aggregation technology to create different types of microtissues in our 96 plates, but then we transfer these microtissues to chips that are interconnected via a common media in our microfluidic channel network. So this chip you see here certainly wasn't our first prototype. Our technologies and platform group have been working on prototypes for over 10 years, mainly in collaboration with institutions such as the Swiss Federal Institute of Technology in Zurich, known to many of us as ETH. So one major point is that the platform uses gravity-based flow on this logically simple orbital shaker that we've designed. Each chip can contain up to 10 microtissues, and they are all connected via the microfluidic channel. So while this gives greater insight to studying tissue-tissue interactions through soluble factors released into the common media, we can also still measure most of the endpoints I mentioned previously. So in fact, we can still image each individual microtissue directly on these chips, or remove microtissues for endpoints such as immunohistochemistry or lytic assays like ATP. So since we've already heard from presentations earlier why there's a need for more advanced human models, I'll just quickly point out a few applications we've worked on with companies, and we have publications on our website that support this. So on the left-hand side, we've worked with a few groups, including a consortium that's public with Genentech and Gilead, on how putting several liver microtissues, represented here by the red dots, in our flow system may better predict clearance of low clearance compounds. Now moving down the line, we've done studies where we can have liver microtissues and tumor microtissues interact. So for one example, we can detect and measure tumor cell killing after incubation of a prodrug that requires CYP-mediated bioactivation, but only if the liver microtissues are included in the chip. And probably the hottest area for us right now, because of our focus on metabolic diseases such as NASH and diabetes, is the interaction between human liver microtissues and human islet microtissues. So we've already well established our human islet microtissue platform years ago. Islet biology and islet-related diseases are another major focus at InSphero. So it made logical sense to build upon both platforms, and we've already optimized conditions so that both islet and liver microtissues can share a common media. That's the first big step. And now we're manipulating the media to represent a healthy person versus a diabetic person. So as you can imagine, there are so many drugs out there for diabetes and NASH alone that may target more than one organ, including adipose, which you see here, that's in the future. So the hope is we can better predict how these drugs will work in vivo by putting them in our more complex models. So with that, I hope I did not go over my allotted time, and I was able to give a very high level view of what is scientifically possible with these small but extremely complex little microtissues. As you can see here, there are so many different groups within InSphero involved in optimizing these models, and we are all interconnected, much like our little MPS chips. And of course, we highly value our external collaborations. These researchers are faced with questions that can't be answered well with in vivo models or simpler in vitro human models. So only together can we try to build upon our models so we can really try to address these more complex scientific questions. So with that, I'd be happy to answer questions you might have.
Awesome. It does look We have one question for you. Celia asks, do you have intestinal systems?
We do not have intestine, but I think IVAL is going tomorrow. They might, I think they're known for their intestine.
Awesome, thank you. Yeah, so at this point, I'll go ahead and Sue, have you unshare your slides, and I'll invite all of the presenters to come back for the roundtable. So this is a time where all of our presenters are able to answer participant questions. So please do ask those in the chat. If they are for a particular company or person, please do direct that to that person in the chat, and otherwise we can kind of ask them all. And I do see I've got one question for Tamaz from earlier, which is, have you ever looked at zonation in your system?
Yes, as I highlight, we do have an oxygen gradient created across our liver microtissues as the media perfuses through them. We don't fully recapitulate zonation of the liver. We can capitulate different areas of the liver by altering the oxygen concentration in the media, but you don't get full zonation across that microtissue. It's not quite big enough. But certainly we can tune that to model different areas of the liver.
Thank you, Tamaz. So now I've got one for all speakers. I'll go ahead and say Tim can answer it first. How do you decide drug concentration and recapitulate in vivo PK in MPS systems? If you're comfortable, Tim, answering that. If not, I can shoot over to another.
Yeah, so we have 10 different tissue models. We haven't gone into the PK/PD dynamics quite as much. It's usually tuning and trying to benchmark with in vivo clinical data that our partners provide. So it's more of just tunability and using our throughput to kind of benchmark that empirically and benchmarking the clinical data. So it's a kind of high level idea.
Awesome. Do any of our speakers have an answer for their systems, or a different answer for their systems on drug concentration? And
yeah, so we already showed that, I showed the one publication we've done with AstraZeneca where we created PK/PD models and we're able to predict their in vivo work. This is Mike Schuller and I have both been working with modeling for 20 or 30 years. So we have developed very sophisticated PK/PD models of our multi-organ systems and are working with places like Simulations Plus, Tabula Rasa Healthcare to integrate our PK/PD MPS model data into their commercially available, clinically relevant PK/PD modeling. And so we can very straightforwardly determine concentrations from our systems that would be applicable for first dose for clinical applications. That's currently, we're working with a number of different companies to be able to do that.
Yeah, I think I'll just tag on to what James said. I think the use of PBPK modeling with these systems is key to get translational readout. It's a really essential part of the puzzle. Something that we also do a lot of work on, both internally and with external parties. And it really depends on where people are in their drug development pipeline, how much information they have about a molecule already from other systems, and what data they can gain. But I know we asked just simple questions like how much does it bind to protein, does it bind to lipids, and so we can take all that information and feed it into those PBPK models to get the right concentration. And then there's obviously the alternative is you can obviously start with a wide range of concentrations to start with. It really depends on where people are in their development life cycle and the kind of questions they want to answer.
Thank you. Other perspective from developers?
I think they covered it quite nice. I think it really depends on what the client has in terms of data before that. But oftentimes if there's no info at all, 10 micromolar is a good place to start.
Yeah, totally. Awesome. Okay. So the next question is for InSphero, so for you, Sue. How big are the spheroids? Number one, do you see necrosis at the center? And how do you ensure that drugs can access the center of the spheroids?
These are all good things that I probably should have covered already. But our microtissues, we keep them very small. And the reason for that is kind of the foundation of spheroids. Anything larger than 300 microns over time will become necrotic because you can't get that proper oxygen penetration into the core of the cell. And so we've done a lot of extensive work on this, and liver microtissues, primary ones, will not divide. So what you seed and forms is what you're going to get. They're not going to grow larger, so you don't have to worry about that, but we keep them less than 300 microns. I would say it's less than 2,000 cells. And that's all the different cell types. We try to keep the ratio physiologically relevant as well, but they do not become necrotic over time. And we can look at obviously LDH leakage to ensure this, but then also we do a lot of immunohistochemistry to cut through the core of the cell. In the microtissue, you can actually see that they're not necrotic. I think there were several parts of that question. Did I get them all?
Yeah, so then the last part was, how do you ensure that drugs can access the center of the spheroids?
Okay, so that is a good question. So obviously with non-small molecule modalities, there are ways to actually, label them like Cy5 or something that doesn't interfere with hepatocyte autofluorescence, but you can actually look over time and see that it's actually getting in there and penetrating into the core. And again, because they're very small, this is not an issue that we've seen with any drugs with penetration. So small molecules, it's not as clear to prove, but the fact is you can see clearance very quickly if large molecules can get in there. If all the uptake transporters like the OATPs are represented, then there's no reason they shouldn't get in.
Awesome. Thanks for that answer, Sue. Other questions that people have for speakers for this roundtable, or comments? We'll take any of it in the chat today. While I'm waiting, I'm going to pull up the final bit of the slide deck as well. Oop, awesome. Okay, we've got another question for InSphero. So first question, we'll split this one up to make it easier. When looking at proteomic endpoints, how do you go about doing this? Do you need to pool spheroids to have enough material for analysis?
Okay, so when we get into omics, that's where it gets kind of crazy. So if people ever ask us for Western blots, we will never do a Western blot, right? You need an entire plate. And so we've looked into omics, and proteomics have come a long way, but I think the transcriptomics were first, right? So as I mentioned, TempoSeq from a company BioSpider has a way that you can do gene expression from a very small amount of material because you're not doing any amplification. You're just simply using cell lysates. With proteomics, we're actually talking to a company, Bionosis, right now, which has ability to, again, use a very small amount of material. And we're hoping, we're working on right now, that you can use just a single microtissue. But of course, we always want to have multiple biological endpoints and represent throughout the entire plate, even though we feel like there's very little variation within a single plate, even along the edge. So I guess that is our approach right now. We are looking into that, but no Westerns.
Great. And then the second question was, when you look to incorporate different spheroids from different organs, how do you determine the number of each of the different spheroids to ensure physiologically relevance?
Sure. So, it's pretty easy when we're doing low clearance because it was only liver. For the tumor liver, I think we did more liver than tumor, but that's a little different, right? Because the tumor microtissues are growing. Now, when it gets into actually representing different organs, so islet and liver, for example, I think what we try to do is think of in the body, how is it represented? So there'd be more liver than islet, for example, but we're still working out those conditions. It really depends on the question you're asking.
Totally. Other questions for speakers? Okay, so while we're waiting, oh, Clive has one. Okay. With so many different models for DILI, are there better for, and this is for everyone, are there better models for early screening and better models for detailed lower throughput screening, i.e., would I start with one company and move to another, or do I need everyone to cover the complexity of this disease? So why don't we go ahead and see, Ben, since you haven't spoken yet in the panel, do you want to start on this question? Gotta unmute first.
There we go. Yeah, sorry, I'm used to Zoom where it was on the bottom left, and I was like, oh no, top right. Yeah, so I think that for when we look at a lot of these different models for DILI, I would say that are there better models for earlier screening than not? I think that, in all honesty, I think how we view ourselves as a high fidelity system. You know, we're really good at answering complex biological questions and really being able to dive into, you know, mechanisms and things like that with compounds. I think that, you know, for us, we're really good at kind of later stage in the pipeline when you have a few drug candidates that you're really wanting to dive in on before you go and for that and you're de-risking before you go on to your clinical trials. I think that's where we see ourselves really striving and really excelling at.
Awesome. Tomas, do you want to chime in?
Yeah, I think there's certainly a place for multiple tools to be used in parallel. I know you've heard this afternoon of, I know there's some high throughput systems, there's multi-organ systems where you have real great levels of complexity. When you bring multiple tissues together, that brings a number of difficult questions around scaling and how do you bring these organs together. And so I definitely think a panel of these, or a suite of these technologies is really going to take a molecule quite efficiently from early stages through to latter stage. We do a lot of work now with companies that don't have thousands of molecules anymore. They use their sort of AI-based approaches to generate just some hundreds of molecules or tens of molecules, and those can go into these advanced systems much more rapidly, generate this translational data much more quickly, and then potentially progress through to the clinic. So there's definitely the direction of travel away from the sort of, I don't know, hundreds of thousands of screens, molecule screens of the past.
Great. James, do you want to chime in?
Yeah, sure. I agree with all that. You know, it really is going to take a combination of providers to really answer all the questions. And you know, that's one of the best things about being in this field right at the moment, which is all of us together can't come close to meeting the demand that's occurring and it's going to occur. You know, so we really shouldn't be competing with each other. And I send people to emulate all the time, you know, for stuff that we can't do. You know, and also for InSphero, we don't do organoids, you know, and so sometimes organoids work better, you know, but I think there's a combination of techniques. You know, what Draper showed could probably do a lot more of your, you know, less involved, but, you know, many screening more compounds. Again, other systems, you know, our systems, some of the others that people have chatted about, you know, can get into the more complex interactions longer term. You know, we can take our systems out for months, you know, and sometimes with DILI that makes it really is very important. You also want to know in terms of DILI at times, you know, whether or not you're affecting other organs or whether other organs are affecting the DILI. This is something that is, you know, we would focus on, and we do with companies. So, you know, but I think really it's going to be a combination. So I think Clive, you're absolutely right that, you know, all of us together, you know, it's going to be 2-3 different companies, you know, really satisfying totally to answer the question you're looking for.
Thank you, James. Tim or Sue, do you have anything to add?
Sure. I mean, I can go real quickly. Sorry, Sue. Obviously, I agree. I think it's a quarter answer that everyone has their advantages, and you sort of have to pick and choose based on what your application is. And even DILI is not just one application. There's not going to be one size fits all model, but you know, consider each model's advantages and disadvantages. So I think again, for a Draper provides, it's sort of a high throughput system as was mentioned. So if you're looking at patient diversity, trying to recapitulate the population diversity, we know that for almost all diseases that there's obviously differential response among different subpopulations of patients. So if that is something that is interested in you, then you sort of need a higher throughput system likely to do that. But you know, so much good biology that has been presented today, you know, showcases the fact that, you know, it's kind of a kid in a candy store in many ways here.
Yeah, thank you. Sue, any final comments for that question?
Well, I think because my background is actually toxicology, I think all of us today speakers, we all face the fact that a lot of companies for like tier 1, tier 2 screening, if they have thousands of compounds, they'll actually use a much simpler model even. They'll use HepG2 cells or primary hepatocytes in 2D. And it's not until they get burned that they want to switch, right? It's easier and it's cheaper. But I think, you know, our company, we actually have been seeing a big movement to, you know, just jumping right into a simple, like a 3D spheroid model can give you a ton of information. It's quite high throughput. And I think, as Timothy mentioned, what you put in also has a huge effect, right? These are primary cells we're all talking about. So if you can use a pool that represents a population for screening, that's your best bet for making sure there's no, you know, idiosyncratic issues with that donor. It's not like a poor 2D6 metabolizer or something. It's a pool that represents the population. Just screen everything in it and see what you get.
Awesome. Thank you, everyone. That's a really good response. And Clive also says thank you. That makes sense to use a suite of human relevant tests that make to pick up the complexity of the family of diseases. So then the next question is a good one. I know this is of interest to the IQ, is to know each company's views and if they can share any ongoing efforts, if any, on building and testing similar liver models for preclinical species, so animal models. Let's start with Timothy.
Yeah, it's a great question. No, we haven't. It's obviously an important consideration to try to validate organ on chip in general, is to benchmark what we know about animals and then hopefully move on to humans, which is obviously a harder problem. I know Emula obviously has some great papers on that, and among other systems here. We don't have that yet. We have not benchmarked much against animals. We have vascular models, which we work with collaborators to benchmark biomarkers with rat and different species and human clinical biomarkers, but not in the liver space.
Great. Ben, do you want to chime in then?
Yeah, sure. So yeah, as Timothy was saying, we have a publication particularly where we were looking at rat and dog liver chip systems, and so being able to use those. We had a publication back in 2019 that was looking at these species models. So we do have that, and that's something that is definitely a key important step in kind of building into as we're all trying to dive into building more models and trying to translate data that's been previously generated to best understand how we can slowly transition and change things over for drug development.
Sue.
Okay, well, gosh, you think I paid the person to ask that question, because actually that's a big initiative that we have right now. We have Sprague Dawley rat, we have beagle dog, we have cyno monkey, and it's probably a tiny fraction of what we work on. 95% of what we do is human-based because we care about human drugs. But the reality is, if someone sees something toxic in vivo rat, but not in vivo dog, what does that mean to humans? And so we're trying to find a way to bridge from that in vivo data to human. And so we do have this consortium, I think I mentioned, it's a cross-species consortium, and there's major pharma in there. So Pfizer, Merck, Takeda, I'm going to forget someone, AbbVie. We're actually presenting on this at SOT for anyone that's interested. But we're really trying to better understand, if you see something in one species, is it because of metabolism? Is it because of transport? What are the issues? So it's not just hepatocytes too. In all of these models, there are also the same strain of NPCs, which is really important for really predicting DILI, for example.
Great. Tomas, do you have anything to?
Yeah, so I think the only thing I'd say, so from our perspective, we've typically focused on human models to predict human responses. We've done a variety of things with other preclinical animal species, rat in particular. We've developed a rat chip. I think one thing that is a challenge for the field is the supply of good quality material from those animal species, particularly primates, and that is becoming a big problem and probably is not easily solved by anyone in this room. And so that is something that people are going to have to deal with, and that's where these tools will become very important. So we can certainly build those models. We have the protocols for doing it, but a bit like Sue, 5% of our work is with human, and so it's not been a big area of focus.
Great. And last but not least, James. do you have anything to chime in?
Right. So actually, I've been around long enough that we started off with animal models. I mean, that's what we published, 10, 20 years ago, and then moved into humans. So we've got lots of experience with animal primaries. We do now currently have 2 NIH grants from NIEHS to build multi-organ animal on a chip models, one for barrier tissues, one for looking at multi-organ tox, and liver is part of that. What we're trying to do is an approach of induced pluripotent stem cells derived, so you get rid of this source issue, because we're pretty good with the primaries. And so what we're trying to do is now we developed some of the tissues from induced pluripotent stem cells from certain animals, but liver is actually really difficult, especially for rat with induced pluripotent stem cells. So even though the primaries are fine, induced pluripotent stem cell from rat is difficult. We're working on that now, but it's anybody has any suggestions, we'd love to hear from it. But yeah, we're currently working on that right now for using NIH funding.
Great. Thanks everyone for chiming in. That was really good to hear from you all. While I'm waiting to see if any other questions come in, I do want to share just a few wrap-up slides from me. So I do want to make sure that all attendees know that we, NA3RC, has an MPS technology hub where you can actually find providers. You can sort by tissue type. So we've got our speakers today and tomorrow, but if you're looking again for liver providers, you can find them. You can sort on that hub. You can find contact information, some publications, their logo, a direct link to their website. So please do check out that. And then I'm excited to announce that our next webinar and workshop pairing is going to be focused on neuro, CNS, both CNS and PNS. We are currently aiming to have the next webinar with 2 speakers at the end of April, and then likely the next workshop at the end of May, right before the MPS World Summit. We're still finalizing dates, finalizing speakers, but I do want to let everybody know to get excited for that as well. You can always visit our website to learn more about NA3RC. You can always email me if you do have a follow-up question. Of course, if you have it for a specific provider, the providers are happy to talk to you, but otherwise, you're welcome to email me. And so those are just my wrap-up slides. Going back to the questions. So for emulates, when comparing sensitivity and specificity of liver chip with spheroids, were the spheroids co-culture or monoculture? And was this for the same donor? That's the first question.
All right, yes. So to my knowledge, they were monoculture, to my knowledge. I can actually double check that though to get back to you on that specific question. But then second part of that, because I happened to see that as well, and was this for the same donor as well? So am I understanding that that question is asking, do we use the spheroids on chip in direct comparison to the spheroids themselves? Is that the question?
Possibly, maybe Sophie can chime in as well.
So I was going to say they were not. So we use primary derived hepatocyte donors. So that's what we were able to use. I think that that's something that we would love to be able to do, to be able to kind of culture when we're comparing organoids to by themselves comparatively to on our chips. That's something that would definitely be of interest. And then.
Second question, yeah, did you compare the 2-cell model versus the 4-cell liver chip model for sensitivity and specificity?
Yes. So in the scope of this, the paper that we were publishing and the data that we generated, we did not compare those two, no. Okay. But that is something that we do have pooled data for some of these other compounds that internally.
Awesome. Awesome. Thank you. Any other questions for our panel? Wait just a couple minutes. Sophie says thank you, so you must have answered her question adequately. And then I am just going to, just in case someone is looking for our Tech Hub, where you can find all of the providers that presented today, as well as the ones tomorrow. I've pasted that in the chat. And I think that if I don't see any other questions, then we may go ahead. Oh, let's see. Do I see a question? Nope, I just see a thumbs up on the link. So you are welcome. Okay. Any final words from our speakers before we sign off, since we've got a few minutes? You can say something or not, it's okay.
No, thanks everybody. It was really interesting to see the latest you're all doing, and you know, and thanks a lot again, Megan, for chairing this.
Awesome. Yeah.
Yeah, I
okay.
agree. It was great to learn more about what's going on in the field. I think we all have a common goal of trying to better in vitro human models for disease and toxicology. So
Yes. Awesome. Okay, well, we will go ahead and let people go 15 minutes early. If you know me, you know I love to end a meeting early. So thank you again to all of our presenters, to all of our participants, and we hope to see you all back again tomorrow. We have the same start time, but we actually officially end 30 minutes early tomorrow because we have one fewer company presenting, but there'll be even more great discussions about liver MPS models. There's such great diversity in the field and lots of exciting technology. So hope to see you all then. And with that, I'm going to end the meeting for all. So thank you and see you tomorrow.
Thanks so much, Megan.
Bye-bye.
Bye.
Thank you. Bye.
Megan will be joining us and will take over from me in probably about half an hour, but I'm going to do the introductions. So welcome to the North American 3 Hours Collaborative and IQ Microphysiological Systems webinar and workshop series, and liver models is our subject. I'll give you a short disclaimer that I have to read out. This webinar and workshop series is made from companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither NA3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. The introduction to the North American 3 Hours Collaborative and IQ and this webinar workshop series. The North American 3 Hours Collaborative mission is to advance science, innovation, and research with animal welfare. We are, without any doubt, very strongly in the area of refine, reduce, as well as replace. Our Microphysiological Systems initiative seeks to increase adoption of and regulatory acceptance of microphysiological systems in coordination with animal studies. We're very much interested in the area of integrated toxicology, where we're considering all tools available to us. The North American 3 Hours Collaborative has 39 institutions, 28 of them are developers in our membership. And as you can see, there's a very wide variety of membership from small to large businesses. We put the FDA there in the middle because it's important that we are connected with the regulatory agencies as well. We are hosting a series of microphysiological system focused webinars and workshops. The webinars are company and system independent and focused on general education about an MPS system or topic. Workshops are company specific and focused on presenting information about commercially available MPS systems. We're going to hear from, you will have heard from 5 companies yesterday and four today. Yesterday we had presentations from CN Bio, Draper, Emulate, Hesperos, and InSphero with a lively discussion at the end. Today we're starting off with InVitro ADMET Laboratories, Discovery Life Sciences, then Lena BioSciences, MicroMatrices, and TissUse. And we'll also again follow with a roundtable discussion. Each company gets 15 minutes in total, including a brief Q&A period. We will be strict on time, so if they use their 15 minutes to talk, that's fine, but they will be stopped. The final roundtable can be used to ask questions of multiple companies or specific companies. So we do ask that you please direct in your question whether it's for a company and put the name of the person or the company, or whether it's to the entire panel. Please submit questions using the chat. Questions will be answered at the end of the webinar as time permits. So please use this to type in. Attendees will be muted and have cameras off. One more thing there, we will all be, let the cameras come on for the presenters for the panel at the end of the session. Hopefully Bonio is here to talk a little bit about from the IQ and then we will move into the companies. Can we start off with our first presenter, Al from InVitro ADMET Laboratories, Discovery Life Sciences?
Good morning, everyone. Good afternoon if you're in Europe or Asia. Good evening. It's my pleasure to present in this wonderful symposium. So I will start out with this premise, that this idea that I think that you have seen many times.
Sorry, Al, you've got the wrong display on. Can you change the display settings?
Oh, okay. I will do that. Better? Yeah.
Fabulous.
Oh, okay. Yes. One of the reasons for the high cost of drug development is the inaccuracy of the prediction of drug properties, right? Most 90% of clinical trials will fail with drug candidates chosen from animals. So I think that Clive mentioned that our objective is the 3R, but I really believe that we're not just 3R, we are also critical to drug development. So the mission of my company, InVitro ADMET Laboratories, which has been recently acquired by Discovery Life Sciences, is to work on this, find ways to accurately predict human clinical drug properties so that we can decrease the failure rate and also provide a way to study drug properties based on mechanism that you cannot that cannot be done easily with our clinical trials. For in vitro system to work, it has to represent the organ that it's supposed to represent, and then of course represent the species it's supposed to represent. So our approach is to isolate the cells from the organs and then preserve them right away. Without passaging, so that the cells will retain in vivo properties. Then, yeah, we all understand cryopreservation is the easiest way to use a cell. You don't have to make the cells, and then you thaw and use. It's the best way, easiest way for experimentation. There are also other advantages as listed here. We first optimize cryopreservation of the hepatocytes. For this workshop is relevant. And we're able to cryopreserve hepatocyte to retain viability, confluency, longevity in culture. Our hepatocytes have beautiful morphology, as can be seen here, and then they can be cultured for like 40 days. And then we can also pool the hepatocytes so that we can have hepatocytes from representing so-called multiple donors, a generic human. Again, they can be cultured for a long time. And we can also, and you have heard from other speakers yesterday, like InSphero, hepatocytes form spheroids very easily, and our hepatocytes easily formed spheroids. So these 999 elite hepatocytes are very useful. And hepatocyte alone is very useful for these major types of studies. But I want to talk about hepatotoxicity, which is well represented by the hepatocyte, right? Because you have the right metabolism, it is the target for drug toxicity, and then you can develop your own, there are lots of physiological relevant endpoints that can be used to accurately represent in vivo hepatotoxicity. When we know that hepatotoxicity is not the only problem, there are many types of toxicity that drug developers run into. How can we predict the other toxicity? Now, you can culture those cell types, but the major problem is that you don't have metabolism. It's like testing toxicity without the liver in the animal or in the human. And that's not right. That won't give you the right answer because metabolism is key to drug toxicity, either metabolic activation, forming toxic metabolites, or metabolic detoxification, making the drug less toxic. Without metabolism, you won't have the right answer. So we need to have metabolism. We need to have the multiple organ interactions like the liver and the non-liver organ. And then we need to use the right endpoint. The MTE requirement, I believe, is very important for any in vitro system. So that's why we come up with the ID mark culture system. This is a system that we have patented in the US and other countries. So the whole idea is that the human body consists of multiple organs that are physically separated or discrete, but they're connected or integrated by the blood circulation. So I want to model this. So look at the this is an example of a 6-well plate, right? You can culture each cell type in each well. And what we have done is to decrease the barrier so that all the wells are now connected. And then, so therefore, if you co-culture all these different cell types, like for instance, you could put hepatocytes here and target cells next to the hepatocytes. Hepatocyte and target cells are separated in different wells, but drug metabolism can occur, and the metabolites can affect the target cell. Very simple system. I keep this low quality picture because this has historical sentimental reason to me. This is my very first picture of my invention. You can see you can have different cell types on the left-hand side as represented by different colors. And then after integration, all the cell types are mixed, but they're physically still separated. So you can after treatment, remove the medium and then look for assay for toxicity in individual wells, so therefore toxicity to specific cell types. We prepared this ID model as a 96-well plate format, compatible with multi-tip pipettes. And we also have them at the 24-well models to give you more materials to work with, like for gene expression. Okay, so this is a wells in a well concept, allowical culturing of different cell types as discrete culture in separate wells, but have them integrated by the overlying medium. Let me show how it works. First, I want to show that, yes, you can add hepatocytes and therefore have metabolism. So we designed this ID model. This is the 4 different chambers. The orange color meaning there's nothing, just medium. And then we have one gray, there's 1 hepatocyte, 2 hepatocytes, and 3 hepatocytes in each well. And you can see that now, obviously, the more hepatocytes we have, the more metabolism we have. That is important, but we need to show that the hepatocytes worked. Sorry. Okay, so then we have to show that we can use the system to measure metabolism-dependent toxicity. We use cyclophosphamide and use 2D3 cells as a target cell, just a proof of concept. It's a cyclophosphamide, which is metabolized to active metabolites, causing toxicity. And this is the different procedures. You guys can get the presentation and see all the different steps. But basically, we treat, and then we, after treatment, we remove the medium and assay using MTT to look for toxicity in each cell type. And you can see that we have 3 different plates. In all different plates, With no hepatocyte, there's no toxicity, very little toxicity with cyclophosphamide up to 5 millimolar. When we add 1 well or 2 wells or 3 wells of hepatocyte, then you start to see increased toxicity. Again, this is what happens in MTT assay. You can see that on top, when there's no hepatocyte, there's no toxicity. When you go down from top to bottom, but at the bottom you have 3 wells of hepatocyte, you can see that all the cells are dead because of the metabolism. So this system can be used to understand what we call metabolism-based classification of toxicity. Now, when you have toxicity, which is the same without hepatocyte or with hepatocyte for all both cell types, for both 333 and hepatocyte, then you know that it's direct acting. The drug is toxic by itself. Type 2 is like cyclophosphamide. Yeah, the metabolite can come out of the hepatocyte, and I'm sorry, type 2 is the one that is toxic only to the cells that's making the metabolite, but it's so toxic that it will kill the cell to make hepatocyte. It won't come out. And this is exemplified by aflatoxin. You can see that only cell hepatocytes are killed, but we do not see the release of metabolites to the non-hepatocytes outside of the hepatocyte well. Type 3 is cyclophosphamide. You can see that the toxic metabolite is toxic to the cell that makes the metabolite, but also can go out and kill cells that are outside of the liver. So this is cyclophosphamide. So this is a very useful way to classify drug toxicity. I want to show them an example where I talked to you about 3D3 cells. Let's talk about something more relevant. I want to give you our study on splenotoxicity using the IDMARC model. We make cryopreserved human splenocytes, and then we show that they're very normal after cryopreservation. This is an example of stimulation by PHA leading to proliferation. This is interesting, right? Cyclophosphamide, a very potent immunotoxicant, totally non-toxic to splenocytes, totally non-toxic up to 4 millimolar. And it's not that the splenocytes are not responsive to immunotoxicants. All the others we tested are toxic, except cyclophosphamide. It's because the other ones are direct acting. Cyclophosphamide requires metabolism. So we did an experiment using the IDMARC model, asking the question, would hepatocyte increase the toxicity of cyclophosphamide to splenocytes? And that's what we found. In a cold culture, we're killing the splenocytes. Yeah. And this is the dose response curve. Yeah. Single culture, only splenocyte, only toxic at 20 millimolar each, just high amount of this drug. With a cold culture, you can see toxicity as low as 0.3 millimolar. Another application of the IDMARC is to look at organ-specific toxicity, and I want to give an example using anti-cancer agents. Right, we cold culture different cell types. Liver, lung, CNS, kidney, aorta, tumor. In tumor, we use MCF7 cells. Then we look for tamoxifen cytotoxicity. You can see that this is dose response curve. Tamoxifen is toxic to all the different cell types, but it's most toxic to MCF7 cells. So therefore, it's an anti-breast cancer agent. And you can come up with the in vitro therapeutic index, right? The EC50 versus the IC50. EC50 is how easily it is to kill the target cell that we want, which is tamoxifen. And then the therapeutic index is IC50 for each organ versus that of the target cell, the tumor cell. And you can see that the therapeutic index of tamoxifen to hepatocyte is the best, but it does have a low therapeutic index to some other organs, like for instance the aortic cells and the kidney cells. Okay, so this is the
3 minutes.
Model. Perfect. Yes. So for the IDMARC model, it allows you to incorporate exogenous metabolism to evaluate metabolism-dependent toxicity. You can use IDMARC to classify toxicants based on the mechanism of the metabolism-dependent toxicity. And also it can be used to look at organ-specific toxicity by co-culturing multiple cell types. It's a real particle system, right? No pumps, no tubings, just a plate. So it's the easiest to use multicellular culturing system. And I believe that is a very complete in vitro model, allowing you to study hepatic metabolism, allowing you to expand it to look at paracrine effect. What's the effect of multiple cell types from the same organ? Endocrine effect. What's the effect of cells from one organ on the property of cells from another organ? And this is something we have already made. We do have a 96 well plate format. The whole plate is interconnected, so that can be used to culture multiple cell types all together to look at the basically a plate representing the human body. All right, this is our ID model. Physically discrete cultures interconnected by the overlying medium, simple, readily adapted co-culturing, allowing you to co-culture multiple cell types. And we have lots of publications with our collaborators, and these are some of those. Okay, and this is our information for our company. With that, I will stop. Thank you very much for your attention.
Thank you, Al. There are no questions have come up at the moment. Please don't forget to use the chat box to put in some questions.
Sure.
But we can easily talk at the end. Please put some questions in there for the end session. And what we'll do is we'll go straight on to Yelena from Lena BioSciences. Thanks, Al.
Sure.
Hi, everyone. Let me see if I can share my screen. Do you all see my screen?
It's all good. It's up there.
Excellent. Good morning, everyone. Thank you for joining us this morning. I'm Yelena Vukasinovic with Lena BioSciences. The purpose of our MPS is to build oxidative and xenobiotic metabolism competence in 3D models. The goal is to restore CYP activity, respiration, energy production, and the activity of active transporters, which depend on the mitochondrial ATP. All this is important for hepatotoxicity, DILI, and DDI. Oxygen is vital for oxidative metabolism. It is the final electron receptor in the mitochondrial electron transport chain, providing the majority of cell energy through oxidative phosphorylation. A cell gets only 2 ADPs by conversion of glucose to pyruvate, 2 ADPs from the TCA cycle, and the majority of energy from OXPHOS. Respiring and energy-producing cells are important for mitochondrial liabilities and toxicity testing. Without oxygen, cells dedifferentiate in vitro, switching to glycolysis. This allows them to evade reactive metabolites and free radicals, misrepresenting potential in vivo toxicity. Drug metabolism confidence requires high CYP activity. Many reactive metabolites are formed by CYP oxidation of pairing compounds. CYPs use oxygen to catalyze over 95% of intracellular oxidation reactions, including the oxidation of drugs. Part of the in vitro challenge is low oxygen solubility in the culture media. Oxygen solubility in blood plasma is also low. Only 2% of tissue demands for oxygen are met by oxygen dissolved in plasma. The remaining 98% is delivered by hemoglobin in our red blood cells. To address this challenge, we developed perfused organ panel MPS, which uses synthetic hemoglobin. The MPS comprises the pump with 2 syringes to perfuse 2 perfusion pile inserts, synthetic hemoglobin, and CDZ scaffolds. The inserts are offered in 48-well and 12-well format. This animation shows the components of the 12-well insert system, which works as any multi-well plate. Media, drugs, and assays are added using ordinary micropipette. And here's how it works. Cells are seeded into CDZ scaffolds and perfused in their own media in different wells. Syringe pump pushes synthetic hemoglobin in and out of the tray to perfuse the cultures and deliver oxygen. Synthetic hemoglobin is twice as dense as water and sits below the medium in each culture well. The wells are independent because synthetic hemoglobin is inert, immiscible with, and insoluble in media, reagents, and drugs, and cannot grow any microorganisms. It dissolves 78 times more oxygen than does the culture medium, and slightly more CO2, but it cannot dissolve more oxygen than what is available in the air, and that oxygen is delivered to the medium on the basal side of cultures. Our MPS models flow and mass transport of the blood plasma components from the perisinusoidal space, the space of these, past the endothelial cells, in and out of the hepatocytes. The pericellular flow rate is low, and we're modeling that with 2 complete up and down strokes in a 24-hour period, corresponding to 16 culture volume exchanges per day. We validated synthetic hemoglobin with cell culture reagents and drugs. It is reusable so long as it is not used with chloroform and heavily halogenated organic compounds, but those are not commonly used in cell culture. To check, add the drug or other reagents on top of synthetic hemoglobin, remove and read the absorbance. To reuse it, sterile filter it. For RNA extractions using chloroform, transfer CDZ to a tube. CDZ scaffold is removable from the plates and transferable to other vessels for imaging using sterile forceps. It is made of glass. The insert system is made in the same plastic as Falcon's HDS, integral insert system. Practical advantage of synthetic hemoglobin is that you need to connect one tube to the pump to perfuse 48 cultures in parallel. The cultures are statistically independent without well-to-well crosstalk. You can check that by adding sterile water to one well and filling the other wells with everything you want to test. In the end, read absorbance of the water, pre and post perfusion. Perfusion is sterile because nothing can grow in our synthetic hemoglobin, and the pH is stable. We measured cellular oxygen consumption rate in our CDZ scaffold and in 2D. Growing cells in CDZ almost doubled the oxygen consumption rate, or the OCR. Then we measured the OCR when medium was below the scaffold without and with synthetic hemoglobin below the media. The oxygen consumption rate doubled when synthetic hemoglobin was underneath the medium, even though the oxygen flux measuring probes were above the scaffold. In 2D, HepG2 cells have 12 times lower OCR than primary human hepatocytes. In our MPS, their oxygen consumption rate increased 4 times compared to 2D. The ROS was the same as in 3D, but glucose remaining in medium was higher, suggesting that the cells were using mitochondria. Respiratory metabolism measured by Alamar Blue is increased in all cell lines we tested. For direct comparisons, the medium volume is the same in all conditions for this and all of our studies. As perfusion pile is an insert and not a multi-well plate, our wells are smaller than those of the corresponding multi-well plate, and our medium column height was larger, as indicated in the parentheses. Larger medium column height limits the amount of oxygen the cells can receive, making our system perform worse than it actually can. But this was the only way to directly compare different conditions. Despite this, in our MPS, respiration doubles at a minimum compared to 3D. Alamar Blue can be reduced by most cytochromes, except for A3. SIPs don't reduce it because their potentials are higher. We like this assay, but what we don't like is that there is a small chance that it could potentially be reduced by conversion of lactate to pyruvate during gluconeogenesis. It cannot be reduced by conversion of pyruvate to lactate, which is what cell lines like to do. Ethanol may also be able to reduce it, but not appreciably without the enzymes. We tested potential Alamar Blue reducers without cells and found that the most likely reducer is NADH or NADPH. We also tested it with enzymes like LDH. Considering the amount of enzyme used and the duration of the test, we're more confident that the majority of the reduction comes from mitochondrial respiration as the assay manufacturers claim. This is how our MPS affects CYP1A1 activity relative to 2D and 3D for various liver cells. Data is normalized to 2D for respective cell types. In our hands, HEPA-RG cells had always lower activity than the primary human hepatocytes then donor pool. iPSCs were about an order of magnitude lower. HepG2 cells had low CYP activity, but their phase 2 sulfation was good. CYP activity in primary human hepatocytes, or PHH, is dependent on the medium used and the extracellular matrix used with our CDC scaffold. For head-to-head comparisons, we always plate the cells at the same time and test them at the same time as the cells in 2D. In CDC scaffold, the CYP activity is the highest with Matrigel. We found that one component in all commercial and commonly used media is too high and practically negates the utility of physiologically relevant oxygen delivery, but helps the cells in 2D early on. PHH in our scaffold take about 2 days to catch up because of that. Having found that CDC does well between days 4 and 7, we checked other CYPs in the 12-well perfusion pile insert on day 7. First, we cultured the cells in 2D and in perfusion pile. Then on the day of assays, we transferred the scaffolds from the perfusion pile insert to a multi-well plate for direct comparison with 2D cultures using the same well size. The baseline activity for 2B6, 3A4, and 2C19 was very high. CYPs are not the only enzymes involved in metabolic activation of compounds. Other oxygen-dependent enzymes like peroxidases and monoamine oxidases participate too. To begin to optimize the medium for oxidative metabolism, we switched to fresh primary mouse hepatocytes. This allows us to take the variables like cryopreservation and mitochondrial integrity out of the equation. Reducing one component of the medium to physiological levels restored oxidative metabolism from the start. Our interest in the medium not only comes from the perspective of oxidative metabolism, but also from erroneous insights we get when looking into the mechanism of toxicity. When we use the medium containing supraphysiological levels of the component in question, we erroneously got high peroxide in 2D. We did not see this when that component of the medium was brought to physiological levels. Regardless of the cell type or medium use, our MPS builds confidence in oxidative metabolism. The question is, how can we really use this to develop better models of mitochondrial toxicity? We trust the primary cells in our MPS, but they are expensive. HepG2 cells cannot tell us if phase 1 metabolite causes mitochondrial toxicity, but they can tell us if the parent can. They're cheap and easy to work with. So we went on to optimize their oxalase, having in mind that the cells are metabolically very plastic and can easily switch to aerobic glycolysis if their mitochondria is in danger. Here's what we found. We can keep HepG2 cells focused on oxidative metabolism. LMR blue is a good indicator if the compound inhibits complexes 1 through 4. For ATP synthase, cell titer glow is more accurate, but it is a terminal assay, so we're optimizing another one. Our ultimate goal is to enable repeat dose testing in perfusion pile. We're early, but the results are encouraging. How can higher OCR in HepG2 cells help us? Here is an example. We tested a chemical toxic to primary cells. It was toxic, but there was something really unusual at a certain dose range that we could not explain. And for some of those doses, the toxicity was practically negligible. We tried many assays. The parent was not interfering with the assays, and we tested the suspect target acellularly with the parent, no inhibition. We did not measure the metabolites, but we didn't think that it was the phase 1 metabolite due to differential response. It was either the parent or a phase 2 metabolite. We tested the parent in HepG2 cells using 2 different media. When the cells were respiring, the chemical was toxic. When the cells were not respiring, they were growth inhibited. The conclusion? Drug metabolism is coupled with metabolism. Using all these models helped us solve the puzzle and identify something new. We hope that you will find the same. Thank you.
Perfect timing there, Yelena. Thank you. We don't have any questions posted yet, so just shouting out to the audience, please remember to post some questions for our presenters, which we can either talk at the end or after each section. So unless there's any questions, we'll move over to Simon from MicroMatrices. Thank you very much, Yelena.
Okay, hello everyone. Today I wanted to tell you about a versatile platform we have developed called MicroMatrices micro-TMA technology, which unlocks the biology of microphysiological tissues. We can use this technology to analyze histopathology, biomarker expression, and transcriptomics across all tissues in the same 96-well plate. The technology can be used for spheroids or organoids from many different tissues, so it is very versatile. Trying to see why my slide doesn't. Oh, okay. Our MicroMatrices micro-TMA technology provides a solution for high-throughput histology screening of spheroids and organoids. It does this by aligning spheroids or microtissues in a single plane on a micro-TMA mold in a format that replicates the layout of a 96-well plate, which is then embedded in paraffin and sectioned onto microscope slides. This approach provides H&E histology data for all spheroids in the 96-well plate, and which can also be reviewed together all on one microscope slide. In addition, we can use parallel sections from the TMA for immunostaining for detection of biomarkers and transcript profiling using TempoSeq technology. As a result of this, we're able to measure a wide variety of endpoints that maximize the value from 3D microtissue experiments. Here's an example of an H&E map derived from a single section of a liver micro-TMA from a cross-species experiment. The top image shows the entire experiment on a single slide, and the lower images are magnified views of individual microtissues showing the level of histopathological resolution we can obtain with the micro-TMA. This allows histopathological confirmation that the microtissues do have the morphology which is consistent of the tissue that they are designed to model. We've used technology to examine toxicologically relevant responses across species at the level of liver and thyroid carcinogenesis in 2 published case studies. In these studies, we've used all the capabilities that I described in the first slide, namely H&E histopathology, multiplex immunostaining, and transcript profiling on parallel slides. We've used MicroMatrices micro-TMA technology to understand the mechanisms which underlie these liver and thyroid carcinogenesis responses. In case study 1, we showed that the technology can define key events in the CAR liver cancer adverse outcome pathway and recapitulate species differences between rat and human at this level. In the second case study, we identified mechanisms which underlie species differences in thyroid carcinogenesis at the level of UGT enzyme induction. And today I'll focus on case study 1. This table represents the layout of a single 96-well plate in a dose-response experiment that was used for the case studies with liver microtissues. For the liver carcinogenesis and thyroid carcinogenesis case studies, we used the canonical CAR activator phenobarbital, which is a rat liver and rat thyroid tumor promoter. And we were able to test rat and human cross-species responses on the same plate. The spheroids were also treated with a positive control for the induction of hepatocyte proliferation, that is, hepatocyte growth factor. Parallel sections were immunofluorescent stain for bromodioxyuridine, a marker of cell proliferation, and images scanned automatically on a confocal scanning microscope. The advantages of this approach for image capture and analysis is that this can be automated on a confocal scanning microscope using a grid overlay such that all the images are automatically annotated with identifiers. And these identifiers can be incorporated into the code of the image analysis algorithm such that the image analysis and data processing can be performed automatically, which avoids any possibility of human error. For this approach, because the image analysis file size is relatively small compared to whole mount Z-stack images, it streamlines the quantitative image analysis process. This shows the image analysis to derive a bromodioxyuridine labeling index using an automated script. The script identifies BRDU-labeled hepatocytes and filters out BRDU-labeled non-parenchymal cells to compute a labeling index for hepatocytes. And the BRDU-labeled hepatocytes are highlighted in the red dot crosshair and unlabeled cells with a green dot crosshair. This shows some example images of the liver microtissues for these experiments for both rat and human liver microtissues. The histograms below show cell proliferation data expressed as a hepatocyte labeling index. We found that hepatocytes in the rat liver microtissues proliferate in response to phenobarb, that's on the right-hand histograms, but those in the human do not. And this was the expected result as it mirrors previously published data that shows using this approach, we can recapitulate known species differences at this level. We also performed toxicogenomics analysis on parallel sections by laser dissecting the liver microtissue sections out of the micro-TMA section mounted on a palm membrane slide. In the published liver and thyroid carcinogenesis case study, we used microarray technology for transcript profiling, but we've since repeated this analysis with TempoSeq with similar results. The process is very efficient because we laser microdissect the spheroid section directly from the micro-TMA into a sticky cap 96-well PCR plate ready for TempoSeq analysis. And the latter process is facilitated using the Zeiss microbeam robo-mover arm, which controls the laser microdissection system. We've used Illumina correlation engine matching analysis to statistically test the match of the rat and human TempoSeq differentially expressed gene data to different adverse outcome pathway signatures for CAR, PPAR, and AHR. The correlation algorithm is sophisticated in that it derives a compound p-value for the match, which takes into account both the statistical significance of the fold changes, but also the polarity of the change. We found that, as expected, there was a significant match with the CAR adverse outcome pathway signature in both rat and human liver microtissues. That's on the top panels on the left and right, but not for the PPAR alpha and AHR AOP signatures. And we were able to show species differences in the lower tables in the induction of UGTs involved in thyroid carcinogenesis, namely UGT2B1 in the rat and in the human, a different UGT, UGT1A6. So to summarize, in case study 1, we've used this approach to recapitulate species differences in key events in the CAR liver carcinogenesis adverse outcome pathway, including hepatocyte cell proliferation and phase 1 enzyme induction. In case study 2, we've shown there is a difference in the induction of UGT2B1 between rat and human liver microtissues that could partly explain species differences in the CAR-mediated process of thyroid carcinogenesis. Using the micro-TMA, we were able to assess histopathology, proliferation, and gene expression changes in a single 96-well plate microphysiological tissue experiment. So this adds value by defining a mechanism of toxicity, facilitating risk assessment that can be applied early in discovery safety assessment. and can also be used to mitigate the impact of adverse outcomes if they occur later in preclinical in vivo testing to support non-human relevance arguments. This approach can also help to validate your 3D microtissue in vitro model by checking alignment with in vivo data in preclinical testing. So I wanted to just re-emphasize the unique advantages of Sphero Matrices micro-TMA. Firstly, our micro-TMA offers an efficient way of reviewing histology or histopathology in H&E stain sections, because the pathologist can review all samples from an experiment on one slide. Secondly, all the sections are stained simultaneously using existing automated staining technology, which is not available for whole mount staining. So there's reduced technical variability and therefore greater accuracy in the data. It overcomes issues of light scatter and antibody penetration encountered in whole mount staining approaches. And the image data file size is smaller, making the data storage and data processing faster. It maximizes value from a single experiment because processing 3D microtissues within a micro-TMA offers the potential of running multiple assays from a single experiment. This is more efficient even than using 2D primary hepatocytes. Okay, so if you're interested to explore this technology, please send me an email. I'm happy to discuss this further with you. Thank you for listening, and I'm happy to answer any questions.
Awesome. Thank you, Simon. My name is Megan. For those of you that may not have met me, I'm the NA3RC program manager, so I'll be helping with the Q&A from now on. So if anyone has any questions for Simon and MicroMatrices, just go ahead and put them in the chat. If we get them in the next couple minutes, we'll answer them now. And if not, then we will move on to our next speaker. Simon, you can go ahead.
Sorry.
We do actually have a question from you. Rita asks, how many cells are the liver microtissues you showed composed of? Oh, you're muted. Sorry.
Sorry, the answer is about 1,000 cells. I mean, the thing about microtissues, generally, that's if one is interested in, I guess, a fair degree of oxidative perfusion or diffusion. They're usually about 250 odd micrometers in diameter because of the limits of oxygen diffusion. So that does limit the size if you want to look at them in that context. I mean, but the answer is about 1,000. So that's the case for many of these microtissues.
Awesome. Thank you, Simon. Any other questions? Okay, well, we will move on to our next and final speaker from TissUse. If Simon, if you want to go ahead and turn off your camera. And then, if anyone has any further questions for Simon or any of the other speakers, you can go ahead and put them in the chat and we'll get them to them in the roundtable. But we will go ahead and turn it over to Eva from TissUse. Thank you.
So first of all, thank you very much for giving me the opportunity to having this talk today. And it does not move forward. Why can't I move to the, okay, it's just quite slow. Okay. So just to give you a brief introduction about TissUse, our company, who we are. We are spun off from the Technical University in Berlin, and we founded 10 years ago. And since then, we've been continuously growing and building a strong customer base. And yeah, since 2 years now, we are even qualified under ISO standards, which is a nice proof of our good quality management system we're having. We are a multi-organ chip company, so this is the technology that we offer. It's composed of one of the chips. There we have several commercially available chips in different sizes. I will go into a bit more detail later on. We can either work with those commercially available chips, co-culturing up to 4 organ equivalents, or we also have the capabilities of doing rapid prototyping, so we can also do custom-made chips. And then there's the devices and accessories to run the chips. We have an active pumping, so it's an active perfusion of media within the circuits of those chips. And this can be controlled in quite a nice range of flow rate and shear stress. And then last but not least, the organ models. We are working with several organ models that also derive from either cell lines, primary cells, or IPS-derived cells. We have our own IPS cell bank where we are differentiating cells. This is also commercially available. And this is the chips that we are offering commercially available. They are sealed from below with a microscopic glass slide, which makes them compatible for live tissue imaging. And they have then the organ compartments. You can see here it's 2 up to 4 organ compartments. Then the size of a 96 or 24 well sized well, which makes them compatible with standardized cell culture inserts like transwells for barrier organ models. The on-chip micropump, which is composed of those 3 little valves in the top, perfuses the medium within these chips, within the microfluidic circuit. So it's a closed microfluidic circuit. We have no external media reservoirs or external media reservoirs. Everything that's in the chip stays on the chip. What we also have developed is a fully automatic system, which is able to completely autonomously run the chips for up to 4 days. It can do all the liquid handling from top and do bright field or fluorescence microscopy from below. It has cold storage for storing the media samples that's taken, or substances or fresh media that needs to be supplied to the chips. So this automated system, it can operate up to 24 chips and at a 24/7 operation mode. And as I've said, it is the media storage capacities. They're designed such as to operate those 24 chips for up to 4 days if we do daily media exchange regimen. And the integrated microscope, it can then not only take the pictures but also analyze and analyze the pictures and use the enormous amount of data generated there for machine learning purposes. So from all this technology, we develop fit for purpose assays, and this means that we prepare organ models. In most cases, the organ models are prepared outside of the chips, so static, normal static cultures, and then they are integrated into the chips. So as I said, we use organ models from cell lines, primary cells, but also can integrate biopsies or tissue slices, as they have these standardized 94/24 well-sized compartments. Then the organ models, they are integrated in the chips. They get used to the continuous flow and the dynamic perfusion. Then we can do a repeated exposure or media exchange sampling, and do online or in-process controls with all the samples repeatedly taken from the system. And at the end of the cultures, retrieve the samples out of the dynamic chip perfusion system and do regular analysis like histology, qPCR, metabolite analysis, omics, everything that you're used to doing with your organ models normally. We have started early with including the liver into those multi-organ co-cultures, as the liver, of course, plays a central role in metabolism and detoxification. And until today, we have established various, well, up to more than 10 of those co-cultures. And yeah, so they are always designed for a specific context of use. So either it's to investigate a second organ toxicity caused by metabolism in the liver, or we include barrier organs, for example, the skin or the lung or the intestine, to investigate the pharmacodynamic profile of drugs or substances and how the absorption through the barrier then influences the metabolite profile of the drug or the substance in those chips. And then we are also moving towards disease models. So yeah, besides those pharmacokinetic and tox content, we have, for example, a co-culture of liver with pancreatic islets, which I will also talk about later, where we are able to study the glucose and insulin regulation by those two organ models. So now coming to the liver models, we work mainly with liver spheroids because they have been shown to improve liver function and prolong stability of hepatocytes over time. We're always aiming for prolonged culture times, and in our experience, those spheroids are widely accepted also by our customers and already used in their laboratories. Yeah, and the spheroids that we produce either in-house from the cell lines or IPS or primary cells in those multi-well plates, forming spheroids of different sizes, or we can also integrate commercially available microtissues, for example, those from InSphero, where we have heard a very beautiful talk yesterday. And then the way we integrate those spheroids into the chip is also various. So either we just have them as directly applied to the glass surface, where they then over time attach or even slightly grow out. If this is not desired for some applications, then we can also have those thermoformed porous membranes included into the chips, where we have separate cavities for each and every spheroid model, and then they have the perfusion below those thermoformed membrane, and yeah, so a very well nutrition of those spheroids. Or we can also have the spheroids integrated into hydrogels, and then having the hydrogels with the spheroids in the chips. And here you see an overview of the different readouts that we can perform. As you can see, the major liver model that we've been working with is the HEPA-RG based liver model, where we use HEPA-RG cells to combine them with human stellate cells at a physiologic ratio, and yeah, these cells, these spheroids work quite nicely in our hands for many applications, and producing albumin at a rate which is comparable also to other systems. Then we have worked with human red primary cells or IPS-derived cells, and of course also, as I said, the primary commercially available models. All those ticks say that we have done those assays repeatedly and have sound data for those, and all those green circles means that it's in theory it's possible to do those analysis, but we've not done it yet, and just because it was not asked for customers. Yeah. And regarding time, I hope I still have enough time. Now I wanted to go over two two examples of what we've done for co-cultures with those organ models. And first, I wanted to show you a project what we've done together with Bayer, looking at the dysregulation of thyroid hormone axis by environmental chemicals. Here we combined a liver model and a thyroid model derived from primary tissues, either of mouse or human origin, and looked if the liver is able to metabolize the thyroid hormones, or if the thyroid is responsive to stimulants, which would then eventually allow us to study the disruption in the crosstalk of those two organ models. First of all, we checked for the markers of the organs for the liver. Of course, it's the albumin and thyroxine binding globulin expression, which is then important for binding the T3 and T4. And we also looked at other markers on gene expression level, and it was quite stable over 21 days. Then for the thyroid model, it could also be cultured functionally over 21 days, maintained in vivo-like follicular architecture, and it de novo synthesized thyroid hormones in a TSH-dependent manner. And in the co-cultures, then we found both glucuronidated and sulfated metabolites in the human co-cultures, while in the mouse setting we had only glucuronidated metabolites. And this is an ongoing work, and in the next steps we want to look into perturbations of this interaction, and also looking at the reduced hormone synthesis by the thyroid model. Next, I wanted to show you what we did together with AstraZeneca, working towards diabetes type 2 model, where we wanted to gain more detailed knowledge on the interplay between the insulin resistance, the pancreatic islet beta cell adaptation, and the progression towards type 2 diabetes. Here we co-cultured pancreatic islands, which we derived from InSphero, and the HepRG-based liver microtissues for up to 15 days. We did repeated glucose tolerance tests. And what we saw is that we could functionally co-culture these primary human pancreatic islet liver spheroids, and that only under the co-culture conditions the glucose uptake and the insulin release was regulated. So without the liver crosstalk, the islets did not show an inhibition of insulin release. So the liver was stimulated by the insulin released from the pancreatic islet to take up the glucose, which then in turn induced a decrease in insulin secretion by the pancreatic islets. And this was stable over the 15-day culture. So we have a 50-day stable culture, and the liver continuously secreted albumin, whereas the pancreatic islets showed an insulin secretion, which slightly decreased in the co-cultures, in the single islet co-cultures, which might be due to the constantly high glucose levels there, and so they lost the functionality. Yeah, the biological process underlying this glucose insulin regulation in the MPS, they're quite complex and nonlinear and involve numerous feedback loops. And therefore, we joined forces to develop the computational modeling, which should describe the glucose metabolism over this 15 days. And therefore, we did a single liver and single or liver islet cultures and exposed them to various glucose concentrations in the media, so either being hyperglycemic, normoglycemic, or hypoglycemic, and used the data we retrieved from those multi-organ chips then to train a model, and there the model then could reproduce the fast variations in glucose and insulin changes also the long-term decline in both glucose tolerance and insulin secretion. So what you see here is that with the daily media exchange, the glucose concentration drops and then peaks up again, and same goes with the insulin. And the over time, over the 15 days, these the glucose consumption decreases, and the last points here that they were modeled then they are predicted by the computational model. So we could predict the short and long-term behavior of this co-culture in the system. And lastly, we used this computational model also to translate the experimental results to the human, which shows a good agreement with published data of the glucose response to meal in healthy subjects. So the 24-hour feeding regimen, let's say, we had in the chip were then transferred to a 4-hour meal human in a complete human setting, and this could be correctly modeled. And with this, I'd like to thank the whole team, and looking forward to having your questions then.
Awesome. Thank you, Eva. So now we will go ahead and move on to the roundtable. So I will go ahead and invite all of our presenters today to go ahead and turn on their cameras, and then just unmute when we actually ask you a question. So I'll actually start. I'll start. There's a couple questions for Eva. So the first question for Eva is, how long can cells be cultured in the humic system, days to weeks? And have any data been generated for ADME gene activity, enzymes, transporters in the hepatocyte model over time?
Yeah. Thanks for the question. We always aim for long-term co-cultures. So normally we do 7 to 14 a day cultures. For many organ models, we have also prolonged the culture up to 4 weeks. For the liver, I must admit that I think we always stop the culture after 14 days, because this is what was then requested from customers. And the second part was the ADME.
Yep. So asking, have any data been generated for ADME gene activity, enzymes, transporter in the hepatocyte model over time?
Yeah. We do have that. So we have done this for the different organ models for either the primary cells, but also for the hepatocyte cells. We did qPCRs and some transcriptomic analysis, and I think most of this data there's also in the publications that I mentioned. Yeah.
Great. And then I know there's been some questions that have both been asked and answered in the chat, but I'm going to go ahead and give the presenters the opportunity to say them verbally, just to make sure that those who are watching the recording can also hear the answers. So I'll actually start with a question for Yelena. Are the seed-easy scaffolds compatible to other cell types, such as renal, proximal tubule epithelial cells, and brain microvascular endothelial cells? Is it possible to measure TEER values of the cells using the seed-easy scaffold?
Thank you for the question. So yes, the answer to the first one, we have plenty of cell types that we tested. Much of that is available online at lenabio.com/cell-types. We have used brain microvascular endothelial cells, and in conjunction with neuron astrocytes, microglia, and I believe pericytes. We have not measured TEER in the scaffold. I think that because the barrier would be 3-dimensional, so the idea would be then the simplest thing probably just to put put the cells in a transfall, use the membrane and measure from there, and you can use our CDC scaffold on top with all the other cell types. In terms of kidney cells, we did not culture those. The only thing that we did, I believe, is kidney cell line HEK293. Thank you. Thank you for the interest.
Okay, so I'll actually then jump to, again, I think Al answered this question in the chat, but I'll give him an opportunity, chance to answer it verbally. In the ID-MOC system, have you observed any issues with media compatibility across cell systems in the various compartments? You're currently muted, so, yep.
Yes, I want to thank Ray Evers for the question. Now that's a real common question. Our approach is this: because we can culture each cell type individually in their own wells, so we plate each cell type in its preferred medium first, right? And then we wait until each cell type is ready to be used. Like, for instance, for some of the butt dividing cell type, make sure they have the right cell density and so on. Then we add the hepatocyte to the hepatocyte wells. For hepatocytes, we let them attach for 4 hours. That's our preference. And then some of our other collaborators, they use to 24 hours. They rather do that, and that's fine too. When each cell type is ready, right? The other cell type is ready in their own media. Hepatocyte is ready in its own media. Then we remove all the media, integrate all the cell type with a common medium. And we use a common serum-free medium, which is the best for treatment, right, without protein binding. And that has worked really well.
Thank you, Al.
Thanks for this question.
Yeah. Then I'll jump back to Eva. Have you explored the HU-MMIC liver chip for liver inflammation type of studies, as well as co-culture with resident macrophages?
Yeah, so that is in theory possible. So we can have macrophages there. For example, the InSphero model, it has some included. So far, honestly, we did not do that yet. We're working towards including immune cells in the microfluidic circuit, and we are also working towards a fully vascularized system, so we can endothelialize all the technical channels of the system. So we have a fully closed endothelial layer covering all the technical channels, and now we're working towards having vessels also so in the organ compartment, so having vascularized models, which would allow us then to have a perfusion of immune cells throughout the whole system, but this is work ongoing.
Great, thank you, Eva. Then we'll jump actually back to Al. In the ID-MOC, was hepatocyte monolayer phenotype augmented, like in other co-culture models, by media sharing with 323 cells? Question from Tom. You're muted again.
The most common statement in the world, you are muted. Yeah, we use primary human hepatocytes, and they're metabolically very active on the day after plating, that's why we use them 4 hours after plating. We don't co-culture with 323. We plate hepatocytes at the cell density that they're confluent, they're stable. So we don't add any other cell types, and we like that the best, because there's no 3D3 cells, right? They may be P450 deficient, but they do have phase 2 enzymes, so we don't want any mouse enzymes metabolizing, complicating the picture.
Great. Thank you, Al. And then jumping back to Eva, David asks Eva to answer the same question about media compatibility concerns in the multi-organ system.
Yeah, thanks. This is a very important question for us because we are co-culturing up to 4 organ models in a closed microfluidic circuit, having one and the same media. So and especially looking for example, if we work with IPS cells, every IPS cell line or differentiated cell type has its very specific media requirements. But so far we always succeeded in finding one common media, which is then a base media deprived of many of the other growth factors needed by all the single organs. And we see that the co-culture it even facilitates the finding of the common medium, because especially if we have liver inside the co-culture, it produces albumin, and it stabilizes the whole system and is supporting homeostatic conditions. Yeah.
Great. So as I wait for more questions to come in, I do want to present just a few wrap-up slides from the NA3RC. So one resource that I want to make sure, and again, feel free to keep typing questions in the chat, but I want to make sure that everyone on this webinar knows that you can use our technology hub to find any of the liver providers, as well as any other types of providers, or even if you're looking for providers who use certain 2 different types of organs, you can sort by tissue type in our MPS technology hub. So that's a really great resource. You also get directed to their webpage, see publications, and just a brief description of the company. And then I do want to announce, again, in case you weren't here yesterday, that our next webinar and workshop pairing will be focused on neurosystems, both CNS and PNS. And we're planning on having the webinar at the end of April, probably like the 26th or the 28th. And then we plan to have the workshop pairing pretty much right before the MPS World Summer, around May, probably 25th, 26th. We're still finalizing the dates and everything, but stay tuned for those, and then we'll move on from there to do other organs. And then I do just want to let everybody know that you can visit our website just generally to find more about our organization, our MPS initiative. There's also even a talk about regulatory status that I presented at the MPS World Summit last summer. And you can always email me if you are interested in joining our MPS initiative. We do have a limited number of end users in our group, and we are open to having more if you are particularly interested or generally becoming one of our sponsors. So yeah, just wanted to let everybody know about that. And then I am going to paste the Tech Hub in the chat. Now I will move back to the questions. Okay, so question for Al from Simon. I'm not sure if that's for Al or Simon. In vivo, there is, I think it might be for Simon. In vivo, there is protein binding.
The question is for Al, yeah.
Oh,
so
for Al. Oh, from Simon. Oh, I'm reading it now. Okay. I see it. Okay. In vivo, there is protein binding of the drug. How do you compensate from the lack of this in your multi-well system to extrapolate data to the human in vivo situation? And yeah, I see you answered it in the chat, but go ahead and answer it verbally.
Great question, Simon. I also have thought a lot about this. So what we do is that we first look at the effect without any protein, right? So there will be no protein binding. We all know that in vivo there is protein binding. So our current approach is we have developed a tissue culture compatible 100% human plasma system. And we have used that to culture, like for instance, hepatocytes, that there will be culture for over 30 days in this compatible plasma. So then we can integrate all the different cell types, especially in the last slide I showed with the human on a chip with all the different cell types. And we use the human plasma, and then we treat with the drug that we want to evaluate. And we haven't published any of these yet, but the results are really promising. Number one, human plasma seems to be compatible with all cell types so far. So we're very excited. Like Eva, you may have some data on that too, and that will be our next integration of the system to make it more in vivo with the multiple cell types and to integrate it with the human plasma. But Simon, that's a great question. I think that that approach will really turn the in vitro system, make it closer and closer to the human in vivo.
Okay, thank you very much, Al. That's great. So you compare with and without and see how it changes and so on. Yeah. Mm-hmm.
Yeah, and then you can, well, because we know the fraction unbound, then we can see if we make sense out of it.
Yes, thank you.
Thanks.
Great. Yeah, no, definitely acceptable. I have questions in between presenters or as well from other NA3RC members. Any other questions for our presenters today during this roundtable? You can always ask them to a certain presenter, or you can ask them to all presenters, depending on what you are interested in. I'll give just a couple more minutes in case someone is typing in the chat. Okay, well, if I don't see anything further, then we may end a little bit early, which if you know me, I love to end a meeting early. We all do. And with that, I'd just like to thank everyone for attending today, for your attention, for your time, for your questions. It's been really exciting these past 2 days to learn about all the different liver models in the field, to learn about, when it might be more appropriate to use one and then another, or have them in conjunction. They all have their strengths, and it's really great to hear from all of these, and we're excited to see how this field develops and how our initiative continues to develop over the next year. I know many of these companies will be at SOT as kind of the next closest upcoming conference. Many of them will be at other conferences throughout the year, and we actually just developed a little tabletop sign for them to display on their booths. So if you are interested in seeking out the companies that are NA3RC initiative members that are devoted to the 3Rs, that are devoted to advancing the field, even working with their competitors to drive advancement, you can look for those little signs. And yeah, Ruth had a question about the recording. So I am going to be just double checking the recording, processing the recording today or tomorrow, and then I will be sending out an email directly to all the NA3RC members and then to our IQ representative, who will then forward it. And so it'll just be a link and a password, and I'll likely leave the recordings active for about a month or so for you to watch. Potentially longer, and that's how you will access them. So yeah, and I've actually already gotten several emails about the recordings of people who missed the live presentation, so we will definitely get those out soon. Awesome.
Will people, sorry Megan to interrupt, will people provide their slides? I mean, we're happy to do that, but maybe that's not, and what is the vehicle if you want to do that? I mean, send it to you, or?
If you want to do that, you can send it to me. I know that in the past, some companies have been more hesitant about actually sharing the slides just because sometimes they're presenting data that's a bit more sensitive. And I will also say, as I said, it'll be behind a password protected wall and only sent to the NA3RC and IQ members. So it's optional if you want to share it, but people will be able to view them via the recording. So
is there a folder or something on the NA3R's website where you can log in and then just go and look for the presentation that you want to see again? No.
We could make that. We can make that probably via our team site versus our website, so it's a bit more protected. Again, you can put a password on.
That's fine. Yeah. Just that's fine.
Yeah. No, thank you, Simon. Thanks for the idea. Anything else from our presenters before I close out? Okay, cool. Well, good to see everyone and hope you all have a great rest of your day and a good week. Bye, everyone.